Hi, Prashant. Can you hear me? Hello, Prashant. Can you hear me?
Yeah. Santosh, you could please start.
We can hear
Sugu,
you.
can you hear me?
Yeah, yeah.
Yes, sir.
Okay.
Yes, clear.
Okay.
Santoshi?
Yes, sir. Yes, sir.
Yeah.
Can we wait for some more time, sir, or we can start?
No, please start.
We shall start. Okay.
Yeah.
Wait 10 minutes. Yeah, very warm good morning to everyone, to all the respected delegates and invited speakers, guests, and dear participants. Welcome to day 2 session in InBix conference. As yesterday we have seen wonderful and scintillating talks, and in the same way we carry forward, like we move forward with the same enthusiasm and with the same active participation of all the participants. So before moving forward, a few glimpses of the today's events. We're going to see, like, we have session 3, biosciences and machine learning. It is followed by panel discussion 2. Then it is followed by keynote speaker 2. Then
Excuse me. Santoshi, there is a
echo sound.
uh, Dr. Ganesh, could you please mute yourself?
Yeah, yes, I can hear.
Yeah, then it is followed by session 4. That is Big's children. Our little kids will perform scintillating talks. They'll give their oral presentations. So last but not least, and the most awaiting and children presentations followed by cultural events. So I request all the participants and everyone to be tuned with the cultural events. So now I would like to invite session three chairs, Kiran K. Telukunda and Sundar Rajan sir, to take over the session 3.
Is the charting disabled?
You could just chat there or something.
Are we going to start now, sir?
Kiran, please unmute yourself.
Or we wait for Sundata?
No, we can go ahead, please.
Okay. Okay, great. Thank you, Santoshi. Yes.
Yes.
Well, very welcome to everybody for the second day and interesting talks. And it's starting with a very interesting talk. It's very great pleasure to me to introduce Dr. Gyanesha Chaubey. He is a population genetist and a renowned anthropology researcher based in BHU, Varanasi. His lab focuses on the migration, drift, and selection of South and Southeast Asian human populations. Apart from several collaborative projects with various national and international centers, his main focus to the identity top 100 diseases of genetic plane and design an ultra-cheap genotype chip to screen them in one reaction are his interest. Apart from this, he has several interesting papers in related to the population genetic structure. So population increase, and during this COVID times, this talk, the contribution of population genetics in COVID-19 research will be a very exciting talk. Thank you, Ganesha. Please go on. Can Ganesha hear me? Dr. Ganesha? Sugu, can you hear me?
Yeah, I can hear you, Dr. Ganesh. Could you please unmute your mic? Dr. Ganesh Chaubey, are you around? I think he'll rejoin.
It looks like he's in, so we will wait for him a couple of minutes.
Yeah, Kiran, I
He's
just
back.
tried contacting him.
He's
Probably
back.
he'll be back,
He's back. He's back.
He's back. He's visible now. Dr. Ganesha, please go on.
So everything is okay, visible?
Yes, you are not visible, not even not the present. Yes, now the presentation. Yes.
Okay, great.
Yeah, please go on.
So very good morning to everyone, and I'm really thankful to Pras for bringing me here. And this is more like a story that people who are working in population genetics, people who are trying to find out the ancestry of certain population, and if their expertise could be involved in pandemic like COVID-19 or some diseases which are widespread all over the world, what could be the outcome and how they can be able to provide their input and their expertise to solve such kind of cases. So as you all know that there was virus, and this virus was creating a different kind of symptoms to different people. So here the case is more like the virus was one, it was changing the variants one by one, but the human was same, and these human were showing a different level of heterogeneity for the virus infection. And this was our main question that when we switched to this research. So let us have some background that what so far we have discovered about a population, about the population genetics of South Asia, and how different populations are interrelated with each other, whether languages are more prominent, whether geography is more prominent in the subcontinent. So let's go through one by one. And most of the cases we have done some bioinformatics analysis, and this bioinformatics analysis is the one where we look different variants, different haplotypes, and slowly we could go one by one to the different analysis which we have done for finding out our ancestry. So if we look to the demography history of India, we see that most of the population, they speak a language which is called Indo-European, whereas the Dravidian population are more restricted to the south. There are some outliers like Gurukh and Oraon. They are present into central and eastern part of India, whereas there is another pockets of language family called Austroasiatic. They are into the central and eastern part, and on the Himalayan fringes there are Tibeto-Burman language family. And interestingly, that most of the linguistic researchers, they say that none of the language family which is being spoken in South Asia is actually originated into South Asia. They have been imported from somewhere else. Whereas recently there are few studies which are showing that Dravidian are indigenous, which has been originated into South India. So if we look to the South Asian population, there are more than 10,000 ethnic groups, around more than 5,000 well-defined population, more than 500 tribes, which are representing around 8.2% of the total population. There are still 36 hunting and gathering populations. As we know that we switch our lifestyle from hunting gathering to the agriculture, and there are still some tribal population who are practicing the hunting and gathering. And if we see just all around us, we can see that we have diversities in language, we have diversity in our social structure, we have different dress and food habit, our marriage practices are quite different in each and every region. And as you know that there is a saying that kos kos pe pani badle, teen kos pe bani. So this is perfectly fit for the Indian populations. And what we do, we study genetic architecture with a background of language, social structure, marriage practices, and phenotypes. So if we look to the South Asian population, where do they fit in the whole world population? This is a principal component analysis. Here you see that there are Europeans, there are Near Eastern population, Caucasian and Central Asian, and then there is a perfect line of South Asian population. On the other side, there are Pakistanis, and to the lower side, you see that there are Dravidian population. And on the other side of principal component, there is East Asian population and Southeast Asian population, the Indian Tibeto-Burman population who are living at the Himalayan fringes. They are more closer to the East and Southeast Asian population, whereas the Indian Austroasiatic population, they are in between East Asian and South Asian population, whereas they are much closer to the South Asian population comparing to the Southeast Asian population. So these are the ancestry overall, where we could see that some of the population they share with the West Eurasians, and some of the population they share with the East Eurasians. And overall, if we look to the geography of South Asia, we see that the most of the population they follow the geographical pattern rather than the language pattern. And here, if we see the more structured Himalayan climb, where we see that the Himalayan population, the population who are living in the Himalayan, they indeed show their closeness with the East Asian population, but they are not following the same climb as East and Southeast Asian population. They are still in between. The South Asian climb is somewhere here, and then there are the population of the Himalayan fringes on the Indian side. They are in between the East Asian climb and the South Asian climb. Then they make their own climb, which is quite unique. And we have also shown that these populations, they have their own ancestry component, which is more widespread. And if we look to the Himalayan component overall in the world, we see that they are more towards east comparing to the western side. So the frequency is much higher at the Himalayan side, and even they are more distributed towards the east and far east. And this ancestry is actually, later we found out that this ancestry is actually wherever it is present, the cases as well as the number of deaths is low for the COVID-19. So if we look at the overall maternal ancestry, even the paternal ancestry also shows the similar pattern. There is a West Eurasian component, there is a South Asian component, and there is a East Eurasian component. So overall, whole South Asian population, they fits into this geographical pattern, where East-West Eurasian populations are carrying their own ancestry, whereas the South Asian populations have their own unique ancestries. So how these ancestries has helped us to determine the COVID-19 distribution. If we look any diseases, there is actually differences, clear-cut differences for a disease geneticist as well as for a population geneticist. As a population geneticist, we look to the overall population, we look various ethnic groups, whereas disease genetics help us just to understand for a single individual. So individual wise ancestry versus the regional or languages or the group wise ancestry. But because the disease which we have focused was more widespread, it was widespread beyond the borders of a country, and this has helped us to understand more on the genetic ancestry, which could help us to increase or reduce the susceptibility against COVID-19. So let us start some information about the COVID-19. As you all know that most of the Indian population were asymptomatic, with some of them are also having mild symptom. So overall the mild symptom and asymptomatic were quite high in India, but there was around 15% of the severe cases and 5% critical cases, and then there are few of them have to go to the ventilator, some of them need oxygen, and some of them also need ICU. But the number of cases was so high, the burden of the disease was so high that these number was also much higher in overall Indian scenario. And on top of that, we have around 80 millions of diabetes 2 population and around 53 millions of cardiovascular diseases, and researchers showed that they have much higher susceptibility comparing to the general population. So India was quite badly hit because of some of the susceptible people and some of the susceptible population. So if we look to the first differences between first and second wave, the second wave was much intense comparing to the first wave. And there was not just a single region. In the first wave, there was strict early lockdown. The virus infectivity was much lower. Public was much more serious. Whereas in the second wave, there was no early lockdown. The virus was more infective. Alpha and Delta variants were much more, around 200% more infective comparing to the previous version, and public was much more careless. When we have done a survey around 1,200 individuals overall into the northern India, we found that majority of the people, they relied on the fake news, they did not take the second wave seriously. So this was one of the major region which has helped for the second wave to rise. And if we look to the virus is evolving day by day, and the virus is making new variants. And what was the region? The region is the virus is under very strong positive selection. We have taken samples from all over the India, whereas some of the other representative world population. And here you see that all of the region has very highly statistically significant positive selection. So this virus is under positive selection, and that is why it is changing their variants. But this is not because of the nature is selecting it. This is because we are helping this to propagate. We are helping this virus because whenever this virus enters to a human cell, it starts to replicate, and these replications usually give rise to the new mutations. And as soon as this virus will stop entering to the human body, this will stop replicating, and that will stop producing any new variant. So our main question when we started to work in the COVID-19, our basic question was why some people they are severely affected and some people they are not at all affected with the COVID-19 even after the infection. So by putting this as a basic question for us and using our own domain, can we identify any ancestry specific genomic haplotype, genomic variant which is associated with the COVID-19 susceptibility? So as soon as the pandemic has started, it was discovered that there is a gene on the X chromosome, ACE2 gene, that is actually the vehicle that is as a gateway, that is working as a gateway for entry of the coronavirus, and possibly where the mutations which are present on this gene may modulate the COVID-19 susceptibility of human population. And the further researches showed that decreased level of this ACE2 expression that mitigates the severity of diseases. So our main question was this because ACE2 haplotypes among South Asian population was not at all known. So we have used the haplotype based analysis because haplotype based analysis, as you may know, that it is much more better comparing to a variant based analysis, a mutation based analysis. Here we use a long stretch of DNA, and this stretch of DNA usually helps because this has several number of mutations, and when these mutations passes from one generation to another generation, if there is a positive selection, there is a reduced diversity, there is a differences which is when we compare to the different continental or subcontinental populations. So we used our own analysis, used never joining method just to reconstruct the tree, FST distances matrices that shows the comparison between two world population. So surprisingly, in our analysis, when we use the whole genome or the whole SNP from the whole 1 to 22 chromosomes, so there we see that the South Asian population always they tend to fall into this category, into this cluster. Whereas for the ACE2 gene, we have found out that the South Asian population was falling unusually into the Southeast Asian mainland, island, and Siberian population cluster. This was quite different story comparing to the general pattern which we observe. So we have also looked to the FST distances, and we could see that the South Asian population, including the Southeast Asian mainland, island, and Siberian population, they show a very strong reduced diversity. So we thought that this is maybe because of there are some region in the genome which is under strong positive selection. So just to in order to understand this positive selection among the world population, we have reconstructed the haplotype based network analysis. So in this haplotype based network analysis, we have named like 3 haplotypes, and these 3 haplotypes are very differently distributed into the world population. So the first haplotype was more prominent into the American and European population, the population who are having more West Eurasian related ancestry, whereas haplotype 2 was moderate and haplotype 3 was more asymmetrically distributed to the East Eurasian related ancestry population, where we see the Siberian, Southeast Asian island and Southeast Asian mainland, whereas around 60% of the South Asian population are falling into this haplotype 3 network. So it means that the South Asian population, they might show the same susceptibility. The majority of the South Asian population, they would show their susceptibility against COVID-19, which will be more related with the East Eurasian population comparing to the West Eurasian population, where we see that the frequency of the South Asian haplotypes are much more lesser comparing to the haplotype 3. And if we look to the geographical distribution of the haplotype, which is distinguished by a very single mutation, that is RS2285666, and this mutation, wherever the frequency of this mutation was high, the number of cases or case fatality rate was low. And when we compared with this case frequency as well as case fatality rate, we found an inverse correlation of this particular haplotype governed by this SNP, and we found out that this haplotype, wherever it was present, the number of cases as well as case fatality rate was lower. And the power of the population genetics was that we have not used any real COVID-19 samples, but because we have used overall the distribution and overall the frequency of this SNP or the haplotype, the people who later have worked on the real population, on the real samples of the COVID-19 positive samples, they exactly found the same results which were so far published into 2 recent publications, where they have found that the allele which we have discovered in our analysis, it was found to be associated with the 2-fold decrease in infection risk as well as 3-fold decrease to develop serious diseases. And not only the ACE2, there are TMPRSS2 gene, which was, as we know, that this is helping for the virus to enter to the host cell, and both of the genes, they are showing a contrasting difference, because the ACE2, as we have seen, the South Asian population, they are clustered with the Southeast Asian population, whereas for the TMPRSS2 gene, the South Asian population are clustered with the West Eurasian population. So this is how we can say that the South Asian population would be moderately in between East and West Eurasian population, and they would show same kind of susceptibility. So based on this analysis, we found out that the ACE2 gene, which are present into South Asian and the other world population, the susceptibility is more related with the East Eurasian comparing to the West Eurasian population, and the haplotype, which is governed by a SNP RS2285666, is being associated with a protective role against COVID-19, and this was lowest among the Maharashtra population and highest among the northeastern state of India. And there was another recent study which was done on the Neanderthal haplotype.
Last minute.
Yeah, so this Neanderthal core haplotype was more present into the South Asian population, and when we have done their phylogenetic relation, we found out that this particular haplotype was not at all showing any association among the South Asian population. And the geographical distribution was also into the same coherence, whereas the runs of homozygosity among Indian population was showed that some of the population who are outliers, they might have a very high susceptibility for the COVID-19. So this is how we have done serosurveillance. This is how we have done some of the catalyst of second wave, and then we found that blood group is also associated with the COVID-19. So, the function and the work of a population geneticist was like showing the association of the population and trying to find out the true association of the population with the COVID-19. And these are some of our publication, some of our press coverage, and thanks to our large number of collaborators from all over India and world, and because of them we could able to trace out, we could able to contribute for this research. So thank you very much for all your attention.
Thank you, Dr. Ganeshwar. That's a very wonderful talk. We are starting with the second day, and I'm sure there will be a lot of questions. So let's start with the first question from Indrani ji. It's an eye-opening session. She would like to know, how did you perform the survey for COVID-19?
The survey, the sero surveys. What does she mean there?
I
The sero surveys
think so,
are
most
the
likely.
genetic survey.
Maybe go with both the ones, because she did not express. Yes, okay, she says yes, sero survey. Yeah.
In sero survey, like we have done two kinds of analysis. Like first, we found out the people who are more exposed, the people who are working directly to the peer-to-peer, directly on the road, people who are rickshaw puller, people who are selling the fruits, vegetables. So, and these people, they were once upon a time infected with the COVID-19, and they also could not identify it. They did not go for the tests. So like in our first survey, we have focused on such kind of people who have like never been diagnosed with the COVID-19, and they have not gone to the severely affected with the COVID-19. So this is like more focusing to the asymptomatic people. And here we have found that around in Varanasi and also the adjoining region, there are around 30 to 40% of the people who were asymptomatic, and they did not know that they have been ever contracted with the virus, but they had the antibodies which was present among them.
So, Dr. Prashant, you want to speak?
No,
or
Kiran, just give room to Sandeep, others. I'll ask later if there is time, please.
Sure. I don't see on the chat right now.
No, it's,
so
he has raised the hand. So Sandeep, you may ask the question,
okay.
please.
Yeah, okay, sorry. Go on, Sandeep. Okay, sorry. Yeah, Sandeep, go on. You want to type or you want to talk?
Okay.
Yeah. Otherwise, in the meantime, Subbu, Kiran. Yeah, Subbu also.
Am I audible, everyone?
Yes, go on, Sandeep.
So my question is, like, we have took the haplotype analysis for this particular study. So is there any other marker apart from RS2285666A66, any other marker which is showing LD with this particular polymorphism, or this is the only one which is causing directly involved in
No.
disease?
Okay, so let us understand it like that. When there is a full haplotype, there are several mutations. So in the full haplotype, there are several mutations, but all the mutations, they are rooted to this RS2285666. So like one, as you know that when there is a single event and after that there are multiple events, that is associated with the first single event. So this is how we understood that possibly, and we are also working further on it, possibly this was the mutation which has happened around 25,000 years back, and there was a strong positive selection on the East and Southeast Asian population and the population which were associated with them. And then there are later mutations which has created this haplotype among all the world population.
Okay. There is a lot of excitement, but the time is limited. I still go with one more question, Dr. Prashant. Please go on. This will be the last question, although I had another 5 questions. Anyway,
Yeah,
Dr. Prashant,
thank you
go on.
so much, Kiran. Always, great hearing you, Gyan. Always a learning experience. Thank you for the wonderful collaboration and learning experience from you. The kind of haplotypes that you have dealt, or we have dealt largely in terms of the COVID picture. There is always, or there could always be a kind of a match for the COVID sequelae or the long COVID symptoms. So, for example, there are a couple of haplotypes that might also show genetic susceptibility to, say, diabetes or systemic lupus erythematosus, as long COVID symptoms are taken into account in the near future. So, do you expect any of these haplotypes to have such minimal genetic susceptibility and association with all other diseases in the future?
Yeah, definitely. There are, if we start to look to the genome, and thanks to the next generation sequencing methodology that has come up in the last 5 years, now we have much more detailed information, and the haplotypes which we could able to see that was not possible 10 years back. And there are few of the haplotypes which are more frequent among the population. The frequency is high, whereas they are also associated with some of the diseases. The explanation for this is possibly there are some balancing selection, like there are some kind of thrifty gene hypothesis also working on some of the Indian population, that when we were once upon a time hunter-gatherer, we switched to the agricultural society, we have abundant food. And then this is also one of the best explanation for the diabetes too. So this is particularly not for the one case. There are several cases where we see that the disease causing haplotypes are also increasing in the population. So this is one of the major concern, and we should revise, or we should look to the hypothesis where we see that all the time the disease causing mutations or disease causing haplotypes, they are reducing their frequency into the population generation by generation, whereas we see a completely opposite pattern in some of the cases. And that could be one of the major research for the Indian population, where we have strong endogamy, the population, several ethnic groups. So this is like never-ending process, but I think one has to start and focus on such kind of questions.
Thank you. Thank you so much.
Thank you, Dr. Gyaneshwar.
Thank you so much, Gyan. Thank you.
Yeah, there's a lot of excitement, but I have to stop here with the questions.
Kiran,
We'll go for,
people can drop any question if they have on my email or anywhere. Thank you.
Please ask on the chat. So I will go for the next talk. Dr. Anshuman, I hope you are ready with your presentation. You can share your screen, Dr. Anshuman. So before that, I would like to introduce Dr. Anshuman. So he's a scientist in Institute of Life Sciences, Bhubaneswar. He holds a PhD from CSR's Central Drug Research India Research Institute in Lucknow. He has also postdoctoral experience from the University of Kansas, USA. His research areas include computational biology and bioinformatics. His lab works on identification of drug targets, roles of RNA in cancer, development of databases, drug discovery, molecular dynamics, and structure-function relationship in protein. Dr. Dixit has more than 76 research articles published in high-impact journals and has also guided 6 PhD students so far. It's a very honor to have you, Dr. Dixit. Please go on with your presentation.
Yeah, thank you very much for a very nice introduction. And I was listening to my previous speaker. It was a fantastic talk. And I would like to thank the organizers to give me an opportunity to talk in such a prestigious event. Today's talk is, can you see my screen?
Not yet. Just a minute.
Just give me a second. Okay, give me a second. I will log out and will re-log in. Maybe there is some.
Okay, sure.
Just a.
Okay.
Can you see my screen here?
Not yet.
Not yet. Okay. What's the problem?
Nagamani Sugnakar, is that any issue, or you are able to see the screen?
It's clear, Parash, clear, Kiran here.
So you're able to see the screen?
No, I'm not able to see the screen, but I'm able to see Anushman Dikshit.
Yeah. Screen sharing is not yet.
Dr. Anushman Dikshit, adjacent to leave, you have share content.
Yeah, open share tray, but it is not showing anything.
Okay. After click on the open share, do you see options of windows coming down? Is it in the Windows IU or in which operating system?
I'm on iMac. Actually, yesterday I had a session. That was no problem.
Okay. Okay.
Or maybe I can send this presentation and.
Yeah,
sure.
Please.
That is also fine.
That is also fine.
Okay, just give me a second. I'm sorry for this interrupt.
No, not additional.
The complete.
Yeah, please, we'll try to share that. Please, not a problem. Thank you. Dr. Anushman Dikshit, to which mail ID you're sending? Could you please confirm that?
I will send it to InBix-2021 at Gmail.
Yeah, perfect. Thank you. Yeah, we'll just present it now. Thank you.
I just sent.
Hello. আহিছা নেকি? Start হলে তুমি আহা মোক বস্তুটো দি যোৱা তাৰপিছত মই হেৰি কৰো। What? Hello.
Sorry for the interruption so far. I have sent this email.
Okay, great. They will be looking into that.
Excuse me, are you able to see now, Dr.
Yes.
Anshuman?
Yes.
Kiran, could you please confirm?
Yes, yes, we can see. Go on, it can be start.
Okay, then. Yeah.
Okay, so we can start. Sorry for the interruption.
Yeah, it's fine.
It's completely unintended. Anyway, so today's talk is about identification of drug response related biomarkers and their potential ligands for the therapeutics of oral cancer. So basically, we know the oral cancer is a huge problem in India, especially in this particular region of Southern Asia, Southeast Asia, where we have a lot of habits that are the known risk factors for oral cancer, such as chewing the tobacco, consumption of alcohol, and chewing of areca nuts. Another problem comes with oral cancer is because it is in a majority of cases, it is diagnosed very late, and there are no targeted therapies available for this particular problem. Therefore, there is the mortality rate with oral cancer is very, very high. Even after starting the conventional chemotherapy, because it is not very much targeted, the chances of success are less, and eventually chemoresistance emerges, which further reduces the chances of survival. Therefore, there are requirements to identify those factors which are related with chemoresistance and which are also related with the survival of the patient. So if we can target the factors which are related with the survival and they are also related with the chemoresistance, these can open a new avenue in the drug discovery for the oral cancer therapeutics, especially for the cases where chemoresistance can be a problem. So before going into the talk, I'll just give you a brief background of the global presence of HNSCC. So this is the sixth most prominent cause of cancer-related casualties. And among the HNSCC cases, the oral squamous cell carcinoma is the predominant type. Especially in India, we will see most of the HNSCC cases are the cases where the involved part is the oral cavity, mostly the tongue, the gingiva, or the buccal palate. The major challenges, as I said earlier, we have three major challenges. The most important is the early detection of primary tumor, prevention of relapse, and the emergence of chemoresistance. So today we will be talking more about emergence of chemoresistance and the strategies to tackle. So this is an ongoing study in our lab, which we'll share with you guys, and definitely I would like to have some kind of inputs so that we can go forward. Can we go to the next slide? Yeah. So this is the general introduction about the oral squamous cell carcinoma. So in the year 2022, the estimated estimated global cases were 0.37 million. So you can understand this is a huge problem. It's a very serious problem. The current treatment modalities include surgery, radiotherapy, and chemotherapy or their combination. So if the cancer is detected early, surgery is the best option. But in later stages, a combination of various modality is tried. The outcome remains poor due to, as I said, lack of targeted therapies as compared to other cancers such as lung cancer or breast cancer, where we have a lot of targeted therapies available. Then the second thing is emergence of chemoresistance to the traditional agents, which is a common problem in almost all of the anti-cancer drugs. Now, a lot of research has been done on chemoresistance, and a huge number of reports have been published. The crux is the cellular and molecular responses to chemotherapy is often a decisive factor. And the chemoresistance is related with the modifications in cell function mechanisms that are mostly linked with the altered gene expression, the methylation, and to some extent the mutation as a result of drug treatment. So the most important thing here is the identification of the factors which are responsible for the chemoresistance as well as the survival of the patients. The important point here is the survival of the patient. So there are certain genes which are known to have significant impact on the survival of the patients. So if these some of these factors may also be related with chemoresistance, so if we can target both of them, we can at least theoretically have a better outcome in OSCC. So the current study was done on the following on these lines, which you can see here on your screen. So the data was taken from the Cancer Genome Atlas (TCGA), and then we have got some data from the NCBI Geo. There are two data sets which we got from NCBI Geo. That is, one is GSE 58074. So where they have done gene expression analysis in response to doxorubicin treatment. And there is another data set where they have taken all the OSCC patients versus the normal control samples. So the first step we did was to identify those genes which were differentially expressed among those data sets. From TCGA, we have especially taken all those cases which are related with tongue squamous cell carcinoma, because the other sets are related with the tongue squamous cell carcinoma cell lines. So therefore, from TCGA, in the HNSCC data sets, we especially extracted the subset which is having TSCC. So we did, in the first step, differential gene expression analysis, and then we tried to identify those genes which are commonly dysregulated in all these three data sets. And then we tried to understand what kind of functions they are involved into. So basically, our goal was to identify those factors which are more related with survival and chemoresistance. So we did a Cox proportional hazard regression analysis, which is used to do this particular analysis is used to do to identify the hazard which is associated with particular factors, especially here in the case those genes which may be related with more hazard, or let's say more deaths. Then we got a large number of differentially expressed genes that may be related with higher hazard. Then we used a machine learning approach to identify a very important feature among them. And then in the same, we did a risk score and survival analysis. So risk score analysis is very important because we can utilize those factors which we have identified using machine learning and Cox hazard to stratify the patients in low risk and high risk. So it is important to have such kind of classifier that can help the physicians the clinicians who are treating the patients, so that they can decide on the treatment modalities, whether they have to go on soft approach or they have to use a combination of multiple therapies. It also helps us to identify those patients with high risk. For that, we can go for some kind of more targeted and under evaluation therapies. So we have, after doing all this gene expression analysis and identification of those genes which are more related with deaths and also are related with chemoresistance, the natural flow was to identify those small molecules, if they can modulate the expression of those particular genes, because our hypothesis is, if the genes are getting dysregulated and if we can identify those molecules that can modulate their expression, maybe we can put things into the in the control, and we can resensitize these drugs to traditional chemotherapeutic agents. So in this particular line, we did structural modeling, because many of the proteins which are related here do not have a PDB structure. And then what we did is, we did 100 nanosecond MD simulation for each of the target. We used 5 targets here in this particular study. So total 500 nanosecond MD simulation. So this MD simulation was done to understand the flexibility of the active site, because that is a very, very important aspect, especially in virtual screening. And then we took the next step, which is the docking of the FDA approved library into these structures. And we took an average docking score in 5 different frames. So as I said, we did 100 nanosecond simulation. So in each 20 nanosecond, we took out 1 frame. So total 5 frames we took for each of the protein, and we docked all those molecules in all of the frames, and the average docking score was taken out. And finally, the hits were selected on various criteria, which also contains the diversity of the hits. So this is the overall methodology. And since I wanted to be focused on more target identification and then the identification of the small molecules, some of the details are omitted because we have a limited amount of time. However, you are encouraged to ask any questions, or you can drop any email if you have other queries. So this is the result of differential gene expression analysis. So here we can see the TCGA and the 2 data sets from NCBI GEO, where you can see the number of differentially expressed genes. So here in the bar plot, you can see the number of differentially expressed genes in TCGA. The genes in green, the genes in blue are the upregulated genes, while the others in yellow are the downregulated ones. So you can see here in TCGA, you will find 2,163 genes which are up, and 1,830 genes which are down, and so on for the other species. And among these 3 data sets, among these 3 data sets, we found there are 168 genes that are commonly dysregulated. So this forms a very significant.
It seems to be very
significant population, because there is geographical diversity. The TCGA samples is mostly Hispanic and other North American samples, while the others one are the Indian samples. So those genes which are commonly dysregulated among all those represents a very, very important fraction. And here a detailed analysis of the differentially expressed genes are shown. So here you can see the innermost string. So this shows the GSE 58074, which is the expression of genes as a result of doxorubicin response, and you will see it is slightly different from the other samples which are non-treated ones. So the other ones are the middle ring is the TCGA. You can see here the genes which are referentially expressed in TCGA, and the other ones is the third GSE data sets where we have non-treated samples. So important thing here is there are certain genes, as I said earlier, which are commonly differentially expressed in or let's say the upregulated in almost all the data sets. So the red ones are showing the upregulated ones, the blue ones are showing the downregulated ones. So if you see, some of the genes are commonly dysregulated in all the data sets, while there are certain genes which are having differential expression among the data sets. So out of this 168, we did a Cox regression analysis, as I said earlier, and out of that we found there are 58 genes among them which have which are significantly related with the overall survival of the patients. So if there are 58 genes, so that was a large number, and therefore we had to do some kind of feature selection method, and we used here a machine learning approach to reduce these number of genes, so that they can be reduced to a more practical numbers, smaller numbers. So next please. Yeah. So after this, we identified five different genes: PLAU, CSK, CSK2, SERPIN1, BOP1, and CCNA2. And if you see here, there are multiple colors. You can see the blue, red, green, magenta, and so on. So the blue one shows the deep deletion. The red one shows the amplification, the green shows the mutations, and so on. So if you see, commonly most of the genes have in majority of the cancers. So here you will see the name of the cancers, and the color shows the kind of changes or the alterations in that particular gene across these cancers. So we have a number of different cancers. And this is the expression data for PLAU, and you will see in most of the cases the amplification can be seen, and that is in the majority of cases, except for the one case in the CCNA2 where we will see the deletion and the mutations are in the majority. So you can say that amplification is the most important reason of these genes, and they are getting upregulated. So if you can identify certain ways to keep their expression in check, maybe we can reverse the or we can at least slow down the further processes. Now we also wanted to see, so we have got to know that okay, these particular genes are altered in multiple cancers, and in majority of the cancers we can see clearly that the amplification is one of the most important regions. In majority of the cases, amplification can be seen. So we wanted to see what happens to these genes, these 5 genes, when we take them to various conditions. The conditions I will show here. So in panel A, we can see the red ones, the red squares show the upregulated genes, the blue squares show the downregulated ones, and the white shows that they do not have significant expression here. So important thing here is, if you look into this panel A, you will see the majority is red squares. So the red squares means these genes are upregulated in majority of the cancers. So these are the cancers, the number of cancers, and these are the genes. So if you look into one particular gene, BOP1, so here you will see it is upregulated in majority of the cancers except one. Similarly, the other one you will see it is upregulated in majority of the cases except one, and so on. So basically, you can say that it is the upregulation of these particular genes are seen in these cancers. And here is the case HNSC, which is the head and neck cancer. So you can see here in all the cases, all the genes are upregulated in the case of HNSC. So this is an important observation, that all of those genes are getting upregulated. That is number one. Number two here is the important the response of these particular genes in terms of survival of those patients of the cancer patients. So if you see here, the red one shows the higher expression, the blue one shows the lower expression of these genes, which are related with the higher hazard ratio. So you can see here these particular genes are in let's say ACC. These three genes are related with higher hazard, or let's say they have a significant impact on the survival of the patients. So here we have seen the expression level that in most of the cancers they are upregulated. Now you will see here what is their impact on the overall survival of the patients in various cancers. And you can see here majority of these genes have very significant impact on the survival of the patients. The red ones show that they have very, very high chances of the adverse chances of having adverse impact, that is the lesser survival of the patients. However, the blue one shows the overexpression of these particular genes are having some kind of protective effect in some cases. So if you see here in a majority of the cases, you will see they have a kind of negative impact. Here is especially the case of HNSC. You can see that majority of them have very, very negative effect on the overall survival of the patients. So now we know that these kind of genes, these selected genes have a negative impact on the survival of the patients. Now coming on to the panel C and D, and these panels are very, very important here in the because
According to the poster,
panel number
says
C
the
shows.
workshop will get over by 1 o'clock, 10 to 1 now. So
Hello.
At max if I finish my segment, the village segment in last.
Can you please mute someone?
Sorry.
Okay,
so
please go on.
The panel C and D, so they are showing the effect of the expression of these effect on this expression of those genes when the cells are sensitive and when the cells are resistant. So if you see here the effect of various drugs, cisplatin, oxyplatin, and doxorubicin and other drugs on sensitive cells, so you will see that in most of the cases the expression of these genes is altered, or let's say when in cancer cases it was high, when we treat them with these drugs, the expression goes down or it becomes insignificant. So here it is important. The red one shows the overexpression, the blue one shows underexpression, the green one shows the expression is significant, statistically significant, but the fold change is not very much, it is less than 2. Here it is not at all significant. The p-value and the fold change both are not significant. So if you see here, you can say that these all of those genes and the expression of those genes go down as a response to the treatment with these drugs. However, in the resistant cell lines, you will see a huge difference in color pattern here. So most of these genes, even after the treatment, they are showing the upregulation of these genes. So you can see here the treatment with gemcitabines in pancreatic cancer cell lines, and you will see 3 of those are showing clear upregulation. Similarly, in many other cases, you will see a clear upregulation in many of these cases. So you can say that these genes might play very important role in the chemoresistance against many drugs, not only one but many drugs. So if we can identify those things, because when you look at the complete picture, that they are their overexpression is related with their overexpression is seen in majority of the cancers. Now they are also related with the survival. They have significant impact on the survival of the patients, and you can also see that their expression in drug sensitive and drug resistant cells is completely different. So that also gives you an idea that they can be very, very important targets for the identification of new therapeutics in OSCC. So having said that, so this was the first part of our study, to identify those genes that can be important for the as a therapeutic options. Yeah, please go to the next one. So then we wanted to see what kind of genes they further interact in the human protein-protein interaction network, and we wanted to see what kind of modules or groups they are involved into, because we know that all of these cellular functions, so they are organized by the protein-protein interactions. So protein-protein interactions are at the heart, because at any given time there could be millions of these protein-protein interactions are going on. The complexes begin, and some proteins come together, they will form a complex, they will do a particular function, and then they will dissociate, and some of them will again associate with other partners, and they will do some other function and dissociate. So these things happen very, very quickly. So if we understand what kind of interaction partners are there, then we can also understand what kind of cumulative function they are involved into. So therefore, we wanted to look into the protein-protein interaction network analysis, and we used a string database for this particular purpose, and we have identified that these particular genes are the network is clearly modular, and you can see clear modules here, 1, 2, and 3, and you can see different processes are there, especially the rRNA processing. So BOP1 is involved with a module that is related with the RNA processing. The other ones are especially the CCNA2 and CKS2, the kinases. So they are obviously related with the cell cycle and mitotic phase, and that's why they are very, very, very important. The third one is other ones, the SERPIN1 and the CLAW. So SERPIN1 is also very, very important because it is important for the dissolution of fibrin clot, and it is also a known target in many fibrotic diseases, especially one pre-malignant oral disease, which is called the oral submucous fibrosis, OSMF, where there is a lot of fibrosis, and there is reduced mouth opening because of the fibrosis. So SERPIN1 is a known target by which we can reduce the fibrosis, and it is also being used as a target in lung fibrosis and kidney fibrosis. So this is a very, very important, and we have found that these particular genes, the selected genes, are involved in very, very different processes. So that was a very important information for us. So that we can use individual molecules, or we can combine those inhibitors together to to target multiple processes simultaneously. Next, please. So now our next part starts. So as I said, some of those inhibitors were some of those protein structures were not available in the protein data bank or elsewhere. So we went into the structural modeling of these proteins. So we used software modeler. And after that, we wanted to see the flexibility of the active site, because that can play a very, very important role in the ligand binding. So we did a 100 nanosecond molecular dynamics analysis on all these proteins. And you can see here this is the RMSD curve, which shows that the simulations are very highly stable. And out of these frames, we have taken one frame at each 20,000 steps. So basically, we took out 5 different frames. So they are representing different conformation of the protein and obviously the active site. So we took out 5 frames for each of the molecule, and then we did our molecular docking using glide docking software, which is a part of Schrodinger's molecular modeling package. And this is showing the identification of hits for BOP1, as I said. So you can see here, these are the molecules, the zinc IDs of these molecules. And you can see here the docking scores. So docking scores are ranging from -8 to -12 here. So we have selected the top molecules. And here, a very, very important observation you can see, some of the molecules have large variation in their binding affinity here. So that also gives us a very, very important clue why we should go for molecular dynamics or multiple conformations, because if you if you dock in this particular conformation, you will see that this molecule is actually very, very good molecule. However, if you go for an ensemble docking, you will see, no, this molecule is not a very good molecule, because in other frames it is not showing very good activity. So it is not consistent. However, if you look into this particular molecule here, this particular molecule is showing very nice activity. The red ones is the better score. So you can say this molecule is much more.
Last 2 minutes, please.
Yeah. So I'll quickly wrap up.
Sure,
so,
thanks.
Yeah. So this is how we selected some of those molecules which are showing consistent binding affinity in multiple frames. And here we have shown one particular molecule, secunavir, which is also a protease inhibitor, which is docking very well in BOP1. So the important thing here is why we have chosen the MPA approved library, because these molecules can be repurposed very quickly and without any clinical problems, because we don't have to go for the clinical trials. And this particular molecule, if you see here, the secunavir, this is also reported to be active in ovarian cancer cell lines in a dose-dependent manner. So this can be a potential hit for our own case as well. Sorry. Similarly, we have another molecule which is called argatroban in PLAU. So you can see here there are different molecules. These are the top hits. And you can see the heat map of docking scores in different frames, and the last one shows the average docking score. So you can say there are some molecules which are consistently binding in different frames, so they should be better hits. And argatroban we have identified. Argatroban is a very, very important molecule, especially very recently in COVID. This particular molecule is used as anti-thrombotic agent or anti-clotting agent, and many of the patients have been given this particular molecule. So basically, what I wanted to say here is this particular molecule, when we looked into the literature, so we found that this particular molecule is reported to be very, very active against breast cancer and melanomas. And so therefore, this particular finding cemented our confidence in our results. The next step will be we are going to test these molecules into this is the last slide. I would very quickly go through it. So if you see here in the multiple steps, these particular genes are controlling different aspects, especially in G1 phase. You will see the BOP1 is playing important role, and if we can block the BOP1 expression, so we can have significant impact on G1. Similarly, in the G2 phase, we can have especially the CKS2 plays very important role, and if we can block CKS2, we can have significant impact on the cellular proliferation. Similarly, in the metastasis, we can also identify here that SERPIN1 is SERPIN and the PLO, they are playing very important roles, and if we can block them, that can also significantly reduce or have significant impact on metastasis. So if you see here, if we can combine these three molecules or two molecules which can block different phases of the cell cycle, that can have a more significant impact on overall outcome of the therapy. And we can also say they can also increase the sensitivity of the existing chemotherapeutic agents. However, these studies are computational so far, and we are going to test these molecules in different cell lines in the coming days. Next, please. Yeah. So this is all for today, and this is our group. And this work was done by Ms. Pratima, who is standing next to me here, and Dr. Subhan Kumar, who is currently doing his postdoctoral research at UCSF San Francisco. And these are the other members of my group. Next. The acknowledgements, obviously, this work was, was not possible without the funding, generous funding from DBT India and the support from the director, DBT ILS Bhubaneswar. And in the last, I would like to thank you all for having a very, very patient hearing, even though we had a small interruption in the beginning. But I would like to thank you from the core of my heart. Thanks.
Thank you, Dr. Dixit. Due to lack of time, we'll take only one question, which is on the chat. Which tool is used to expression analysis?
We have used GPR2 for the gene expression analysis, and for GO2R in NCBI for gene expression analysis for geo data sets. For TCGA, we have used GPR2. Okay,
Veena, probably you got answer. Thanks, Dr. Dixit. We go for the next talk. That will be from you, Devapriya Kumar. While he joins us, I would like to introduce him. Professor Devapriya Kumar has received his PhD from Pondicherry University in Indian Institute of Chemical Technology, followed by a postdoc in the University of Maryland at Baltimore. He's currently a professor at International Institute of Information Technology and heads Center for Computational Natural Sciences and Bioinformatics. He currently is a project director of iHub Data, a technology innovation hub, and head of INAI, an applied AI initiative for healthcare. Currently, his group is applying machine learning techniques for molecular design and healthcare. He has been a recipient of awards such as Chemical Research Society of India Medal, Chemical Society of Japan Distinguished Lectureship, Google Impact Scholar Award, Indian National Science Academy Young Scientist Medal, JSPS Invitation Fellowship, and Innovative Young Biotechnologist Award. That's a very huge profile, and it's a very great honor to host you, Professor Devapriya Kumar. You can go on with your presentation.
Yeah, thank you very much for your kind introduction. You can hear me well, and then you can see my slides, right?
Yes, please go on. Thank you very much.
Yeah, thank you. So thanks for having me in this nice meeting. So today, what I thought I will do is to give a brief overview of how artificial intelligence methods and the modern machine learning techniques have come into large use in molecular design in general, and then I will give some examples of some work where we could apply it in the drug discovery scenario, right? Okay, so originally, scientific research was thought to be of three different paradigms. First is the experimental sciences, where we discover new things, or we try to understand naturally occurring phenomena using certain experiments. Right. So this has been there forever. And in terms of theory, so we observe or we make certain observations in experiments, and we try to come up with laws and theory that kind of explains these observations, right? So that's the second paradigm. And then the third paradigm is the computational sciences or computational natural sciences, where we apply these theory that we develop, and then basically develop new tools that apply this theory, and then this leads to faster discovery of molecules, materials, and also making the experiments more efficient, right? So if you look at in the last 2 decades or so, the fourth paradigm that has come in is the data-driven science, right? So you have large amount of data coming from experiments, and data coming from computations, and possible data that can be derived from the theory, right? So you have large amount of data, and then can we use this data to advance science in a way that computations have been doing, right? But slightly possibly better, right? So this picture kind of tells the kind of explosion or the exponential surge in these areas that's happening in the last especially in the last 3 years, right? So this basically plots the number of research articles just in the ACS journals, with machine learning as a phrase appearing in the abstract, right? As you can see, it's a huge, surge in the recent past, in terms of how we apply machine learning in fundamental sciences in general, but in this case, chemistry, right? So, machine learning, or the modern machine learning methods, are being applied now at different scales. We can, for example, look at, the high energy physics, or, quantum mechanical energies, for example, of atoms and molecules, and then looking at sequences of larger proteins and, DNA and RNA. And then, at multiple levels, we are applying machine learning, and then, discovering new ways of, doing science, right? Okay, so just to quickly go over why we started doing this, right? Excuse me. So traditionally, our research group at IIIT Hyderabad has been working on applying computational techniques like statistical mechanics, classical mechanics, quantum mechanics to study chemical and biological processes, right? So one of the things, and I just take this example where we look at the equilibrium of the protein in its unfolded state, in how this equilibrium is affected with respect to external perturbations. This is one of the projects, or this is one of the problems that we have been working for a reasonably long time, right? So in this case, what we do is we run really large time-scale molecular dynamics simulations using different enhanced sampling techniques. For example, in this case, what we show is the free energy of the protein unfolding. When you go from left to the right, the protein is unfolding, and then how this free energy profile changes with respect to different perturbations, right? So from there, we go back, and then we could do free energy calculations or quantum mechanical calculations, and then try and understand the underlying physical phenomena that is affecting this particular process, right? So from then, of course, we did we extended this to understand more phenomena, biological processes, in terms of how these interactions play a role, like, for example, transporters, modified DNAs, protein ligand interaction, and so on, right? So now, there is a fundamental issue in what I just explained, is that when we do really long time molecular dynamic simulations, especially involving structures which are very, very different, right? So, for example, if you take this is an unfolded RNA. If you take this one, this is a completely, this is a folded RNA, and this is a completely unfolded RNA, right? So, normally what we would do to understand these trajectories is to come up with certain order parameters, and then based on those order parameters, we try and understand the pathways and thermodynamics and so on, right? So there is a bias that we introduce as a chemist. We use our chemical wisdom and then come up with these order parameters, but this need not be correct, right? So we started working on a problem about 4 years ago, trying to see if it is possible to start from an extremely complex scenario where the molecule is, moving along, let's say, thousands of degrees of freedom, and come up with a very simple qualitative understanding of, let's say, have the initial state of the molecule, final state of the molecule, and how, it goes from here to here. What are the different intermediates it's visiting? Which ones are more probable, and then what is the kinetics and things like that, without having to define an order parameter, right? So I'm not going to go into the details, but then, we, used unsupervised machine learning techniques to start from here, and then, predict, qualitative kinetic properties with respect to this particular system. So this is how we started, looking at, machine learning as a tool to apply to chemical systems, right? So because, there are a lot of students, I'll quickly, go over what is the fundamental difference between the traditional computational methods we do versus the machine learning way of doing things, right? So traditionally, what do we do? Let us say I'm trying to do a quantum mechanical calculation, or let's say, I'm trying to do a docking or any such, traditional computational method, right? So usually we start from one molecule or multiple molecules, and then represent these molecules in a way that I can, my computer program can understand. For example, this could be the atomic numbers and the nuclear positions, right? Or, Cartesian coordinates, internal coordinates, right? Any of them, right? And then we have a program or a software which, implements a certain theory. For example, Gaussian 16 is a software that implements the quantum mechanical methods, right? A large class of quantum mechanical methods and density function theory methods. So now what is that we are interested in? What we are trying to do is calculate a property y, which is as a function of x. In other words, x is known, which is the molecule that we are starting with, and then f is known, which is the fundamental physics behind these programs. And then what we are interested in is y as a function of x. So when you go to machine learning, so we start from a data set, right? So what do we mean by we start from a data set? That for a given input x, y is known for a large number of cases, right? That's basically what a data set is, right? So in other words, what we are trying to do here is, x is known and y is known for a set of molecules. Okay, so what we are interested in is to evaluate this f, or estimate this function f. So how do we do it? We start from the same input x, and then we use our chemical wisdom, or a chemist or a biologist would sit and then translates this x into a form which is understandable, or which is in some sense machine learning friendly, right? So, and then now I know, corresponding to these x and x primes, I know exactly what the y is for certain number of compounds. Now, what do? We know y, and we know x prime because we know x. What we are interested in is finding f. So that's where, most of the times we use some kind of neural network, which is considered as a universal approximator, where we train with, by mapping x prime to y using this neural network. So now once we have trained this function and we are happy with the performance in terms of this function being able to reproduce all the known y's, we can now take this and plug it in here. We basically replace the software that we traditionally use, replace this software by the neural network, and then we can start to predict properties of new systems. Right. So, I'm going to give a couple examples of this particular problem where I have an input, and then we do what is called a featureization, and then we train for a neural network to predict the properties, right? So one example is, calculation or estimation of electronic energies of small molecules. What is the idea? Idea is that, we use quantum mechanics to achieve high accuracy when we model molecules, right? But beyond a certain size of a system, we cannot handle these systems using quantum mechanics, and hence we switch to molecular mechanics. For example, proteins, DNA, RNA, and so on, right? So we switch to molecular mechanics, but at the cost of reduced accuracy. So now the idea is that can we come up with neural network potentials that are as, efficient as molecular mechanics in terms of computational time, but then they move closer to quantum mechanics in terms of accuracy, right? So if you remember from last slide, I spoke about X, X prime, and F, right? And Y. Okay, so this is basically X prime, right? This is my X, let's say that my molecule is X. Now we have to featureize the molecule in a way a machine learning model can be developed, right? So, this is a scheme that we developed wherein any given molecule whose three-dimensional structure is known, we represent them by a list of bonds, angles, non-bonds, and dihedrals, which are basically feature vectors which are different dimensions. So now we go here. So as I said, this is my X, and this is my X prime coming from this particular scheme, right? And this is my Y. What is that we want? I am trying to train this neural network to make sure that when I start from a given X, I get the true value of Y, right? So these are some of the results where this plot gives you the relative energies of some C11H22 isomers, different classes of molecules. And this is the ground truth coming from the density functional theory calculations. And this is basically the predictions coming from the neural network that was developed using using this particular molecular representation, right? So just for reference, this is a semi-empirical quantum mechanical method, just to show that the neural network potential does reasonably well compared to even methods which take more computation time. This is one example. Second example I'm showing here is how is it possible to apply this to molecular dynamic simulations, right? So the idea is that this is a simple liquid organ system where we try to see if we can use machine learning to improve the accuracy of the simulations that we otherwise do with, let us say, Lennard-Jones potentials, right? So, if I take a, organ system, right, liquid organ, we would normally apply a Lennard-Jones potential as the force field, and then we would do the simulation. So let us say that that's basically the green one here. So let us say, we are able to do some high-level DFT simulations, right? So obviously, in terms of the computational expense, it's going to be very, very high, but then we will possibly reach, or we will move closer to the experimental values than the molecular mechanics or the Lennard-Jones potential, right? So here the idea is what we said was, okay, we know how to calculate this, right? And then this is something that we can calculate for smaller systems, but not really for larger systems. And then hence, we will take the difference between the two, right? And difference between the two, and then, basically, predict the difference in the forces, right? We use forces in the molecular dynamics simulations, right? So this force right here. So we'll take the difference between the two and then model that using the machine learning, right? So that way you will do a machine learning, molecular mechanics calculation plus a simple feed-forward pass from the trained neural network, and then add these two and then do a simulation. So in terms of computational time, we are still closer to molecular mechanics, but in terms of accuracy, we do very well. That's basically what we show here, and we showed it on multiple number of properties. And recently, we applied this to a similar method to calculating, modeling, metal clusters. Right. So, and we can also extend this to, for example, biochemical properties, right? For example, if we have a protein, right, and then if I am mutating a given residue to some other residue, right? Now I'm interested in understanding what is the change in the folding free energy, let's say, right? So, basically, that's what we did. In this case, we defined the environment of this particular residue in the wild type and the mutant, and then based on the environments, we train a neural network potential to predict the difference in the difference of free energies. Right. So similarly, we also recently developed an algorithm wherein for any given protein structure, we predict the binding pocket and then also segment the pocket shapes using a CNN model. So what is common among all the 3 or 4 things that I explained is this. So that, we started from an X and then we do a featureization and then we train a potential to do a certain prediction, right? But nowadays you see that people are talking about deep learning, methods, right? What is the difference between deep learning and machine learning? Basically, you don't really use your expertise to create featureizations, but you let the network itself what the important features are from your original input for doing a certain prediction, right? So basically, the networks become far more deeper than here, right? So this is especially useful when we have large amounts of data available, right? So in this case, this is my original input, right? So, and then there is a network which learns or automatically generate features corresponding to this original input, which is goes into the prediction network to make the prediction, and hence this becomes Fy equals F of Z, which is the featureization space that is learned by the deep learning model itself. Can you please tell me how much more time I have approximately?
So you have another 7 minutes. Sorry, 6 minutes.
Oh, that's it. Okay. All right. So, just to give an example of how deep learning method works. So in this case, what we have done is we developed a chemically interpretable graph interaction network to predict solvation free energies of any drug-like molecules, right? Let's say I have a drug-like molecule and then I have a organic solvent. What is it that we want? I want to calculate the free energy of solvation corresponding to the solute in this particular solvent, right? So in this case, we represent these as graphs, and then we have 3 different phases that we define as the message passing phase, interaction phase, and the prediction phase, using large deep neural network which is trained end to end, and it makes the prediction of the solvation free energies. As you can see, this is the predicted versus the ground truth. And the most interesting part in this case, as I said, this is chemically interpretable in the sense that we take this particular interaction map, and then that interaction map seems to explain in terms of why we get a certain number in terms of favorable interactions and unfavorable interactions, and we showed it on multiple systems with this interpretability or the explainability works reasonably well. Okay, so I'm going to take a couple of minutes and explain this particular slide, right? So today, when we talk about drug discovery, right? So what is it that we are interested in? So if you look at the estimates of the number of drug-like molecules that are possible, there are estimates ranging from 10 power 23 to 10 power 60, right? So what is that we're interested in? That's among those 10 power 23 to 10 power 60 molecules. This is basically the space that that comprises these molecules, right? The overall chemical space. So we are interested in 1 or 2 molecules for a certain target, right? But then this is the known chemical space. What do you mean by known chemical space? That these are the molecules that have been synthesized at least once, right? So these are the things that we already know, and these are the space that we do not know, right? But then, in the recent past, if you have seen, for example, the Zinc database and things like that, this has become really large in the order of, 100 billions, right? So, but then, if you want to do calculations on billions of compounds, that's not going to be possible, right? We are limited by our own budgets, whether it's experimental or computational time budget or monetary budget and so on. And hence what we do, we end up um, sampling a smaller space. Even when we do docking, we are not going to do docking on billion compounds, right? We are going to possibly do maybe tens of millions. So this is basically the sampling space. So what does it mean? We end up only identifying, let's assume the cross marks are the molecules that we are interested in, right? But then this is the only space we are sampling, and we will end up identifying only these compounds. And we will completely miss out on these compounds because we are not sampling there. And also, how about molecules in this particular space, right? So in the next two, work that I'm going to explain, I'm going to see and try and convince that, even with a sample size of this much, it is possible to cover all these top hit molecules when we apply machine learning. And then when using the information that is available here, we can actually we can do a focused exploration of the rest of the chemical space. Okay, so this is the first work where we go from this space to identify all of them, right? So in this case, what we do is, we use Bayesian optimization. Just to, I will not go into the details because of the lack of time. But then the idea is that, we perform docking explicitly only for about 5 to 6% of the whole of the database that is available. But then at the end, we will recover all the 100% of all the hits in this thing. I'll just show this plot. So let us assume that I have 100 million compounds, and then I'm sampling 5% of the space, right? In terms of the top hits, I'm going to only get about 5 to 6% of the top samples that you're going to hit. But then the algorithm that we developed, which uses the Gaussian process and expected improvement, we can see that by sampling only about 6% of whole of the data set, we are able to recover more than 95% of all the top hits in all of the database, right? So this can be also extended to multiple objectives, for example, the docking score and then the log P. And if this is the region of interest, we just start sampling molecules in this region only. And we don't have to worry about, this particular space because we are not interested in. Right. So, and then the second problem is this, that, this is what we have been talking so far, right? So once I have an input, we train an F to get an output, right? Now the question is, can we ask the question reverse, saying, can we map Y to X? In other words, can I simply ask a question saying, I want a molecule which has a binding affinity of a certain value with respect to a given target, instead of asking how much binding affinity this particular molecule have with respect to a given target. We ask the question other way around, right? That's basically a new class of problems called the inverse problems, right? So I will just, I think I'll stop with two more slides. First, let us think of a pipeline, right? So, is it possible to identify molecules directly going in this direction with desired properties? Once the molecule has been identified, can we, have an algorithm which will calculate or which will tell us how to actually make that particular compound? And then the third one is, can this input can go into a robotic synthetic platform, and then that can start doing the synthesis. And once the molecule synthesis is done, that is fed to the spectrometers, and then the spectra comes out. Once you have the spectra, can you go back and ask, corresponding to the spectra, what is the molecule, right? So all these are examples of, these three are examples of inverse problems. I'll just give one example and then I'll start. in this case, what we are interested in is to generate molecules which have a certain binding affinity, right? So, we have what is called a generator. That's a neural network, which basically understands the chemical language and starts generating new molecules. And then you have a predictor. It could be a neural network or a computational calculation or, an experiment, which will evaluate the property that is of our interest of the molecules coming out of here. And then you have reinforcement learning pipeline, which basically looks at the properties coming out of here and then rewards or penalizes this particular generator depending on what the value of the property is, right? So depending on that, we can generate molecules which have specific properties that we are interested in, right? So similarly, you can also, start from an NMR spectra and predict the structure using reinforcement algorithm, right? So I will just quickly summarize saying, the modern machine learning methods are seen to be emerging as a disruptive class of methods, and they seem to have started making important contributions not only in technology areas like computer vision and natural language processing and so on, but also in fundamental sciences like in molecular sciences. And also this is turning out to be an extremely powerful addition to a computational chemist toolkit. What I tried to do today was to give some examples of different classes of problems that we can, conceive and solve using machine learning. So, in our group, we've been working on different methods, and these are some of the algorithms that we have developed, which are relevant to the drug design process, right? Okay, so with that, I will, thank all the students and collaborators and coworkers who were working, in all of these projects and funding from different agencies, both government and private agencies towards some of our projects. And thank you really very much for your attention. Thank you.
Thank you, Professor Devapriya Kumar. That was a wonderful talk. We have we have a lot of questions. Let's see how many questions we can take. The first question is accelerated drug design based on the stepwise algorithm?
That's it? Is that the question?
Yeah.
Okay. I don't know what you meant by stepwise algorithm. I'll use this slide to explain, right? So when we take a complete drug discovery process, right, it starts with the disease and understanding the biological pathways, the corresponding to the disease, all the way up to bringing the drug to the market, right? So, if you look at some of the papers that have come out in the last 3 years, you will find that, the data-driven methods have, been shown to have potential to participate in any of these tasks, right? So starting from the diagnostics of the disease or, the target identification, drug repurposing, structure prediction, you name it, you have, it's possible to, there are, the data-driven methods have a lot of potential, right? So when we talk about drug design in the chemistry sense, we are in this particular region, right? So if you look at some of the methods that we have developed, they contribute to different tasks within this, right? So, the methods that I spoke about are different tasks, right? So the question is, instead of doing it stepwise, can we combine them and do a complete pipeline? Maybe it's possible. I think people are moving towards that, where things are all automated. That's again one of the slides I tried to show here, right? If you look at this, that's exactly what is happening. So when you start, this, but if we are able to develop algorithms which will take care of this whole pipeline, we just have to tell the algorithm that I want a molecule which will bind to SARS-CoV-2 main protease, let's say, right? That's all I need to tell the algorithm, right? So this algorithm should go end to end in terms of the synthesis and characterization and give us a physical compound, right? So, and then, if possibly that can be automated further to evaluate the biochemical assays or and things like that. So it is possible to make it a long efficient pipeline. I hope I answered
Yes.
what you intended to ask.
Yes. Thank you, Professor Devkumar. There are actually several questions, but due to time, we can't take more questions. Maybe that will be possible in the buffer time. So we go to the next talk, oral talks. For the oral talks, it is 5 minutes. And please be on the time, and you will be reminded 1 minute before that time the oral talk is getting finished. Sundata, please go on with that.
Hi all. First, we will have a lecture from Prabina Kumar, Meher and Anush Kumar on SPRO predicting multi-level abiotic stress responsive protein in plant using machine learning algorithm. Yeah. Can you start the lecture, please, Prabina Kumar? You have 5 minutes to complete, and we have 1 or 2 questions for 2 minutes. Are you ready?
Is Rohit Shukla ready? Rohit Shukla and Tirthraj Singh?
Yes, sir.
Okay, please go on. Probably you can go. We'll start with the second talk, and then if the other person comes, we will switch over the time.
Thank you. Sir, can you see my screen?
Not yet. Yes, please. Now we can.
Okay. Thank you, sir.
Good
morning, sir. Myself, Rohit Shukla. I'm research scholar.
You can put it in the presentation. Sorry. Yeah.
Sir, I put it in presentation mode.
Oh, okay. No, it's not visible still like that.
Okay. Now, sir?
No, not yet. Do you have multiple screens?
Yes, sir.
Yeah, maybe you have to revert the screens.
Okay.
So that's. Well, if you can't do it, go on with that. If you, yeah. Okay.
So now it is visible?
Yes, it is visible. Yeah. Yeah. Go
on. Good morning, sir. Myself, Rohit Shukla. I'm research scholar in Department of Biotechnology and Bioinformatics. Today I am presenting entitled Identification of AD-associated genes using machine learning techniques. So AD is generally diagnosed in the late stages due to limitations in the currently available diagnostic methods. The AD is mainly diagnosed by the plaques and tangles accumulation in the brain, and which is done by the MRI, CT scan and other methods. So the currently available NGS-based methods are generating a lot of useful information, means a lot of genes which can be classified as the AD and non-AD genes, and that genes can be provided to the in vitro and in vivo researchers to further validate the things. So keeping in mind, we have developed a machine learning based method which can distinguish between the AD and non-AD genes and can sort the genes for further study. So this is our comprehensive methodology. We have retrieved the AD associated genes from the DigGNet dataset, and then we have removed the redundancy by using the CD hit. We have removed all the duplicate sequences, partial sequences means less than 50 sequence, less than 50 amino acid, and then we have finally have the 1086 sequence for the AD associated genes and for the non-AD associated genes, and that dataset is divided into the 80% training dataset and 20% test dataset. After that, we have calculated the compositional based features and network based features, and then we have plotted that features using the TSNE method into 2 dimensions. And then after plotting, we have implemented the light GBM based feature selection method, Riper feature selection methods, and that selected features were employed for the 16 machine learning algorithms, including several ensemble classifiers. After that, we did the hyperparameter tuning, and then we have finally validated our model, and then our model is predicting the probability score with the AD and with the non-AD. So if we are giving the input, so it can predict this type of result. So this is the feature calculation. We have calculated 22 different type of features with the 13,504 dimensions, And these are the various feature encoding schemes like amino acid composition, grouped amino acid composition. We have also calculated the network based features, because the amino acid composition is the same for the AD genes and for the non-AD genes, the genes which are involved in the Parkinson disease and other disease. So the network connectivity is different between the disease gene and between the non-disease gene, or between two different diseases. This is the feature contribution analysis. We have firstly plotted all the data into the two features dimension, and we have seen that our data is not able to classify between the AD and non-AD genes, because all the points are overlap between each other. So for that reason, we have selected 16 machine learning algorithms which are widely used. That are the ensemble classifier, that are the tree-based methods, that are the linear algorithms, and that are the other methods. And after that, we have applied these 16 ML algorithms against all 22 features, and we have seen that these are the features. All the 21 features, like sequence-based features, are showing approximate only 50 to 52% accuracy, but we got the accuracy more than 90% for the network-based features. So from here, we have found that network-based features can perform best as compared to sequence-based features. So this is the highlighted accuracy of the network-based features. So here we have seen that CAD boost based method is performing best with the 0.92% accuracy, with greater than 90% accuracy. After that, we have applied the two-step feature selection approach using the light GBM algorithm. We have wrapped the light GBM algorithms, and after that, we have applied all those features, all those 22 feature including schemes. So this is the original size of the features, and this is the reduced dimension of the feature. So it has selected one by one feature, and then it has predicted that, okay, out of 20 features, 9 features are important and they are not correlated with each other, and they are showing the good accuracy with the 52.43%. So here we have seen 2 to 3% increase in the accuracy, but we have seen 2% accuracy increase in the network-based feature with the 94.33%. So after that, we have seen that, okay, the two-step feature selection approach is also not increasing the more accuracy for the sequence-based features. So here you can see that this one is the network-based feature accuracy, and here we have seen that the ensemble classifier and tree-based classifiers are performing good for the network-based features, while all other linear classifiers are not performing good. This is the color, and this is the color representing the accuracy. After that, we did the feature fusion analysis, because this is the major approach in the biological analysis. So we have fused the features like AAC and DPC from the original feature, and we made 187 dimensional features. After that, we did the optimal feature size. So 168 is the optimal feature size, and it has increased the accuracy approximate 10%. So here you can see 66% accuracy is increased. So we have applied this is the graphical representation of the accuracy from all 16 algorithms, and we have seen that it has increased the 60% accuracy for maximum features. So we have selected all those features which are showing greater than 60% accuracy, and we have applied the hyperparameter tuning here. In the hyperparameter tuning, we have seen approximate 70% accuracy for the CKSAAP PAC feature. So we have seen that sequence-based features cannot perform good as compared to network-based features. So for the network-based features, we have selected 5 machine learning algorithms, and we have selected the features. So we have found that, okay, light GBM-based 4 feature dimension is performing good as compared to other features. So here we have selected 4 features, and we have applied the hyperparameter tuning. So without parameter tuning, the accuracy is 94.87%, and with the parameter tuning, the accuracy is 97.23%. For the test set also, the accuracy is increased, and these are the hyperparameters, and these are the sensitivity, specificity, and other statistical parameters. The conclusion, we have selected 13,504 features, and then we have applied a rigorous algorithm. We have applied two-step feature selection approach, and for the sequence-based features, we have not found the accuracy greater than 70%. But for the network-based features, we have found the 95%, more than 95% accuracy from the CAD boost method. So we have proposed the CAD boost method for the identification of AD-associated genes, and these are the selected references. And thanks for listening. Thanks for your questions. And sorry for the time, sir.
Yes, great. So any questions? Yeah, there is one question. Which are those hyperparameters that were used to train the model, and how much differences was obtained in terms of accuracy or AUC? What all ensemble algorithm was used? Can you name a few?
Yes, sir. So these are the hyperparameters. Like for the depth, we have increased from the 1 to 100. For the iteration, we have increased from 1 to 500 with the 50 interval. And for L2 underscore LR, this is the we have increased from 1 to 5. And when we have tuned the parameters, so you can see here, this is the without parameter tuning accuracy 94.87%. And this is the train parameter. So the accuracy is increased approximate 3%, 97.23%. So for that, for 3 algorithms, we have increased the parameters here. We have increased the different parameters. So these are the hyperparameters, and this is the 3% increase in the accuracy for training set, and these are the 2% accuracy in the test set. And we have also seen, we have for the parameter increase, hyperparameter tuning, we have only calculated the accuracy. So with the 96.55% accuracy, the sensitivity, specificity, precision, AUROC, and MCC is also good for our proposed machine learning method.
Great, Rohit. That was really wonderful talk and a nice answer.
Thank you so much, sir.
Yeah. So we go for the next one.
Thank you, sir.
Next is Anushree, right? Anushree, Paisha, are you ready?
Yes, sir, I'm ready, sir.
Go on.
Banushree and Natha Roy will present their in silico screening of potent bioactive compounds from Manuka honey as an alternative drug to control SARS-CoV-2. Here is you can start. Yeah.
Yes, sir. My presentation is visible, sir?
Yes.
Okay,
yeah.
Thank you, sir. Yeah. Myself, Vanushree Vaishya, research scholar, Department of Zoology, Mizoram University, presenting my piece of work on in silico screening of potent bioactive compounds from Manuka honey as an alternative drug to control SARS-CoV-2. So as we all know that honey has been used as a food and medicinal product since ancient time. It is rich in antioxidant and antimicrobial properties. So in the present study, we have selected Manuka honey, which is a monofloral honey, which is derived from the tree Manuka, and scientifically which is known as Leptospermum scorperium. So at present, the deadly disease SARS-CoV-2 is creating havoc among the human communities. Until now, no specific cure or antiviral treatment is developed against this disease. So in this present study, we have selected 50 Manuka honey compounds and 4 conventional drug compounds to investigate their interaction against 7 different SARS-CoV-2 related target receptors. So these are the objectives. The first objective is to find out the bioactive compounds in Manuka honey and to investigate their pharmacokinetics and drug likeness properties. Comparison of Manuka honey compounds with 4 selected conventional drugs against SARS-CoV-2 target enzyme by molecular docking study. So this is the workflow of our present study. So at first we have done screening to select the possible target receptor of SARS-CoV-2 for the interaction with our ligand. So we have retrieved the protein ligand or protein receptor from the RCSB PDB, and after that we have done screening and selection of bioactive compounds from Manuka honey, selection of conventional SARS-CoV-2 drugs, and after that we have done retrieval of ligand from the PubChem online database, assessment of pharmacokinetic and admit properties using mole inspiration and switch admit online server, and finally protein small ligand docking using Padstock and Firedock online free server. So this is a detail about our methodology. So these are the list of receptors and the ligand molecules that we have chosen for the present study. So we have selected 7 receptors of SARS-CoV-2 target and ligand molecules, a total of 19 ligand molecules, which is consisting of Manuka honey bioactive compounds, 6 phenolic acid compounds, and 9 flavonate compounds, and 4 SARS-CoV-2 conventional drug molecules, which are listed here. So these are the 3-dimensional structure of the prepared SARS-CoV-2 target receptors, which was done by using Chimera Bioinformatics tools. So these are the 2-dimensional structure of the ligand molecule, which are derived from PubChem database. So we have done the amino acid composition profile of the receptor molecules, and we have found that glycine and threonine are the dominant amino acids, which is with a percentage of 10.8 among the all receptor molecules. So after performing our docking study with Patchdock and FireDock server, we have listed according to the receptors top 4 results of our docking study with most negative global energy. So these are the list of different receptors with their highest docking scores of the ligand. So these are the 7 receptors with their docking results, ligand docking style. So after that, we have done screening to find out the best phyto compounds from Manuka honey. And among all the phyto compounds of the Manuka honey, we have found that quercetin has shown good binding with the S protein receptor of the SARS-CoV-19 with a binding affinity of minus 51.95 kilocalorie per mole. And after that, we have done an whole screening, and we have found that remdesivir is a commercial drug, has shown best binding affinity with the SARS-CoV S protein receptor with a binding energy of minus 57.28 kilocalorie per mole. Yeah, okay, sir. So these are the representation of 15 best docking results, which are based upon their global energy. So these are different colors of this graphical representation shows the different receptor molecules. So we have seen that S protein has shown good binding on the remdesivir, followed by the TMPRSS2, which has shown best binding with the Comstat mesylate, which is a conventional drug of SARS-CoV-2. After that, we have found that S protein receptor has shown good binding with quercetin, followed by different other molecules. So in under this 15 base result, we have found that 7 base position has been occupied by the phyto compounds of honey, and 8 position has been occupied by the commercial drugs. So these are the after finding the best result, best phyto constituent, we have done phyto pharmacokinetic characterization of the ligand molecule, and we have found that phytoconstituent have followed the Lipinski's rule, but the remdesivir commercial drug has shown 2 violations to the Lipinski's rule. So after that, we have done drug likeliness properties analysis using Switch ADME online tool. So at last, we can conclude that most of the honey bioactive compounds from honey has shown good binding affinities towards the SARS-CoV-2 receptor molecules, and honey products and constituent may provide effective ligand for controlling SARS-CoV-2 and may be valuable in the search for COVID-19 therapeutics. It is expected that the finding of this work will be able to contribute a significant and major therapeutic impact in the pharmaceutical and nutraceutical companies. And further, this is a computational study, so further validation of the this particular study is needed. So for that, in vitro and in vivo analysis for in the future days is necessary. So at last, I would like to acknowledge Department of Geology, Mizoram University, for providing necessary infrastructure facilities to carry out this research work. And these are some references that I have cited in my presentation. Thank you, everyone.
It was a good work. And any questions from the audience? One question I have, did you use different type of honey or a specific number of set of honey used for this test?
No, sir. For this particular study, we have selected a particular type of honey, that is Manuka honey, which is a monofloral honey. So from that particular honey, we have selected different phenolic and flavonoid compounds for the particular docking study, sir, to see the interaction with the receptor of SARS-CoV-2.
Because a particular honey may be not available everywhere, right? Because it is going to be a good
Yes,
product if it works, combat of
yes,
the corona. So did you try with other honey which is available in other places?
So, sir, till now, we have not tried with other honey. So we have selected this Manuka honey as in literature. We have found that this particular honey is having more antioxidant and antimicrobial capacities. So we have selected this honey for this time. So in future, we think I think we can go for other kind of honeys too, sir.
Yeah, that's good, because that will be useful, because if it specific a particular thing, it may not be available everywhere in the world, because now everyone is very interesting thing. Okay, thank you. Any other question from audience?
Thank you, sir.
No, we can go for the poster talk.
Okay, thank you, sir.
Thank you. Divya Jindal and Manisha Singh, are you ready?
Yes, sir.
So you have 3 minutes.
So
Yeah,
can I
start
share my
presentation.
presentation? Sir, is my screen visible?
Not yet. Yes, now it is. You can make it in the presentation.
Oh.
Yes.
Yes.
Is it possible to make
Good morning,
more
everyone. This is
larger?
Divya from JP Institute of Information Technology.
Can you make it more larger, maybe?
Yeah. Yes. Okay. Good morning, everyone. This is Divya from JP Institute of Information Technology. Today, I'll be presenting. I'll be giving you the presentation on my poster with the title "Computational Validation by Exploring the Drugs Involved in Alzheimer's Disease, Potential Contagious Omnifera," which is ashwagandha. So, at the beginning of the third millennium, due to the prolonged aging and the new developmental disorders, they are growing at a much higher rate, and there is a much deeper knowledge of the brain which is necessary. Scientific and the technological researches from molecular to the behavioral levels, they have been carried out in many different places. If we talk about their demographic data, in 1 billion people, nearly 1 in 6 of the world's population, they suffer from the neurological disorders, taking it from Alzheimer's, Parkinson's, strokes, multiple sclerosis, and many more. So I'll be covering up the Alzheimer's disease. It is a progressive neurologic disorder that causes the brain to shrink, which leads to the atrophy and the brain cells to die. Out of 55 million people, around 47 million people, they suffer from Alzheimer's. Earlier, it used to at a very old age, around 50 to 60, but nowadays it may set for the age of 50. For AD, there are various that have been designed and came into the market since 1968, but in total, 7 drugs came into the market. That is the Meda, Marplan, Donepezil, Galantamine, or Memantine. In 2021, there is one more antibody which has been introduced, which is Edulam. These all drugs, they have efficacy of 90%, but they somehow cause the toxicity, or some drugs, they are unable to blood-brain barrier. Some of them, they have a low GI absorption, and some they associate with vegan. So this shows the non-specific behavior of these drugs. Technology or the alternative, which is less toxic and are very much enriched with bioactives, showing the neuroprotective activity. So for our study, we have we choose Vithania somnifera, which is ashwagandha, and Indian ginseng. And for this experiment, we have taken 6 proteins. One is ApoE, which is apolipoprotein E, acetylcholinesterase. Third is butyrylcholinesterase. Fourth, base one. Fifth, NMG, and MOA. MAOA, and I have studies using glide. So basically, it's to the FDA approved drugs which I have mentioned earlier. And these are the docking scores, like for the highlighted ones, the bold ones are the
last 15 seconds
for each of the proteins. And from the result, it is very evident that affinity greater than the controls, and from these results it is very clear that the quercetin, it is have binding with almost all the compounds. Rest, if we talk about their admit properties, they all follow all the rules and to be a potent drug candidate. And if we talk about the amino acid involved, they contribute to the same amino acid as the amino acid can contribute by the control. And these are the references. Also, I would like to acknowledge JP platform who present my Thank you.
Thank you. Thank you.
It was method. Can I ask a question? Did you use any algorithm, any specific, any algorithm like neural network or anything? How you validate
No,
these
sir.
domains?
Basically, sorry.
Yeah, Divya?
Sir, your voice is breaking. Yes, sir.
For validation.
So these
For
studies
computational,
still need the further validation. We haven't done the MD simulation yet. So these studies still need further validation by MD simulation or in vitro studies.
Okay, so it is a future work. And
Yes,
okay,
sir,
any
we
other?
are in collaboration with.
Okay, yeah. That will be interesting because unless you do by neural network or kind of stuff, right, you cannot confirm things are happening in the proper direction. Couple of algorithms.
Sir, if you talk about these
vector,
targets which I've taken, they somehow interrelated to each other.
I think we can offline take a discussion on that. Yeah, because time is now very short. Other session.
Yeah. Yeah.
Kiran, this is the last session,
Yes.
right?
Yes, this was the last session. So yeah, with the lack of time, we have to skip the buffer time for networking, and we will directly go for the panel discussion. So I'll hand over to the panel discussion panels.
We can go on. Kiran, are you taking up?
Let's thank the chairpersons, Kiran and Sundar, with a big thumping round of applause, and also to the speakers who has taken a wonderful day two as a wonderful start. Yeah, so thank you so much. So thank you, Kiran and Sundar, and thank you all the speakers. We missed one oral presentation, but I've requested Praveen and Anuj to convert that into a video abstract. Yeah, thank you so much. Yeah. So, assuming that all the panelists are here, let me just cross-check, please. Yeah. Narayana Shastri sir is not there. We have Harpreet, we have Kiran, we have Sona Charles, I believe. Yes, Sona Charles, and Gareth, will be joining later. And yeah, so we will start with this. So the main reason behind starting this is we have what you call Uday here, who is accompanying me for this panel discussion. We both are the report chairs, and we have some elite guests. I hope Dr. Pardasarthi is here. Uday.
Yeah, I think he's there.
Yeah. So Dr. Pardasarthi was a wonderful speaker yesterday from IITR. So he's also there. We have Harpreet Singh, who is a professor at Ansraj Mahila Vidyalaya. We have Kiran Telukunta, who is the founder of TMS Foundation. We have S. Nagamani, who is a scientist at CSR NEAST. And we have Sona Charles, who was again a wonderful speaker from Galaxy Workshop. And Gareth Price, again, another speaker from Galaxy Workshop. He represents, Queensland Center for Computational Facility over there in Australia. So I just, I'm sorry for the background noise. I apologize for this. I'm in an office room where we have some open offices. So the background and key stakeholders here is that, ever since Galaxy has started, we thought of having a kind of a Galaxy India enterprise. So for which BioClues has been a stakeholder ever since. TMS Foundation, from which Kiran has really been taking leaps and bounds. And we have CSR thanks to Narhari Shastri and Viday Mahanta and of course S. Nagamani. And ever since we thought of having what you call Galaxy hub or a Galaxy server from India. We thought of having a kind of a spoke at Hansraj Mahila Vidyalaya because that's where Harpreet is there and Purnima are there. And Gareth has really been kind enough to put a sub-instance in their Australian as well as European servers so that we can acclimatize. Thanks to Kiran for this.
That's a bit correction, Prash.
Yeah, that's
that's from
right?
the Bjorn. That's a bit correction. The Galaxy project.eu is from Bjorn Gruning, from the European.
Yeah, it's what. I'm sorry. Yeah, that was I forgot. Yeah, and of course, a full support from Galaxy project has been there. And together over the last 1 year, 3 months, we have organized about 7 workshops and over 800 from India have been benefited. And there are of course challenges. We are going with a thumb rule called Together We Can. So keeping in view of this, we have some questions to the panelists. So Hriday and I will take you to some of the questions. So to start with, we'll probably start with a small teaser. So the teaser here is with Sona Chalice. So Sona, I know you have started your career as a researcher, you did your PhD. Now you are a group leader. You have your own fellows. You have started using Galaxy as a kind of an enterprise. How good do you think is a kind of a requirement for Galaxy India node? And how did you and your students take this particular Galaxy over the last few years? Yeah, thank you.
Yeah, so it's been almost 1 year since I joined the Indian Institute of Spices Research. So here, so much transcriptomics work, then metagenomics work is going on, especially in the case of plants and with the plant pathogens. So what I found was most of the students were from the biotechnology background. So they require a kind of assistance when they are dealing with the huge genomes and the very large FAST files. So they require a lot of assistance, which cannot be obtained on a single day over programming platforms. So in that case, I believe that they need some kind of initial training using the Galaxy platform, which is actually very easy to use. They have all the tutorials and they will get a lot of benefits out of using Galaxy. So for a beginner, I would recommend them to use Galaxy platform for their initial analysis. So I believe there are a lot of students all over India who would be needing such a platform. And the sad part is many are still not aware of whatever resources they are having. So here I have introduced Galaxy to so many students and they are using it and they are very comfortable in using Galaxy. So that is my observation from here. And especially if you take the plant genomics part, not many are into it, but still we need a lot of attention in this area.
Right, wonderful. So Kiran and Harpreet, do you have any quick say on this, please?
So I have questions. So Sona, you said that you've been informing or let's say telling others. So what are the methods or modes you have been using? Let's say, is it about training or how is it about going on?
So initially, I would recommend them to use the tutorials that are already available from Galaxy. So I'm planning to conduct trainings also, but not very soon, maybe next year or so, because so many people require Galaxy. And if they are not knowing anything about it, I think a training is a must, especially for the people who are doing their research in plants.
And how was your experience with them?
Yeah, it was good. I mean, see, if we are recommending some other algorithms or softwares, I would see students coming around all over me, asking me, "Ma'am, I got stuck in this step. I want your help." But with the case of Galaxy, that is not there. About which I'm really happy because they are getting the gist of what Galaxy is trying to tell them, like, you can use this tool, you can just do whatever you want using Galaxy. If you get an error also, you can just report it directly to the Galaxy server and the people will respond to them. So this is kind of wonderful if you ask me, because they are running into errors and they are trying to clear it up by themselves. So this is actually really good.
Yeah. Harpreet, do you have any quick say, please? Yeah. Please restrict your points to only 30 seconds, please. Yeah. Thank you.
Yes, thank you very much. So I must say that Galaxy is a wonderful platform, as Sona said, that people struggle in using and we are not aware of. And in Punjab, I have an additional problem that even you need to run Galaxy, you have some background data, you must have some background hypothesis, and it require high-end bioinformatics. So even people here, they are not into that level of bioinformatics. So the first challenge is to bring them to that level of bioinformatics, and then also to make them aware that you can use Galaxy to analyze your data, and you can get a lot of things out of Galaxy. So I have a dual challenge. So what we are trying to do is I'm working at both ends. I'm asking my research groups or the collaborators and others to make more dimensions, explore new dimensions of analysis, do more bioinformatics and use Galaxy also. So by using Galaxy, I'm making aware of people why Galaxy is useful, when Galaxy should be applied, and how they can be used. So to answer the question of how, we are trying to conduct many workshops, small workshops. And in fact, I will say that the first Galaxy workshop in NGS, it was conducted in 2014 with the help of the Galaxy people and Dr. Sonika Tyagi. She was there in Australian Genome Research Foundation then. So in 2014 only, we took a step of introducing Galaxy to the people to do NGS analysis. And since then, there was a gap of some year, but a couple of years back, we are again on the track, and we are very much accelerating towards making Galaxy as a, people use Galaxy.
Yeah, thank you. Thank you so much, Harpreet. Dr. Pardasardi, do you have any quick say, please, on how your students have really taken into consideration or cognizance of Galaxy?
Yeah. Thank you. Thank the organizers for this this kind of a thought-provoking platform, discussing about how we can expand this Galaxy at India. So the Galaxy per se, like, it's really a great platform, as all of you agree upon, like already highlighted on that issue. The main thing, also many of you also, we all aware of it, like one thing is popularization and training, and the kind of work it's coming out of our research group also need to be prioritized or push it forward, like AlphaFold claiming, like many drug discovery work I have seen, like it's using Galaxy. They are like published in excellent journal, and it's getting huge popularization, particularly in scientific community. And not only the scientific community, you have to reach the college and university level, like wherever they are teaching bioinformatics, they should get aware of this platform. They can get all resources in one place, as well as like the open source resources, particularly in one place, that will enable them to utilize this more often, and that can also helpful for them to fine tune themselves towards their taking the path in research. And only chemoinformatics is not only that, like we currently expanding into toxicology and other issues, like how we can do disease biology using Galaxy platform. That's what currently we are focusing in our research group. But main thing lacking is popularization and the training to the students, reaching at various stages, researchers and colleges, universities, and the hosting locations. Like not only that, like if many people started using it, probably you need the intensive resources also. That's also one need to have some plan for how we have to expand this Galaxy network. Yeah, that's it. Thank you.
Wonderful, wonderful. Thank you so much. The next 2 points I would request Hriday and Nagamani as key stakeholders, because this is something what I thought where we could discuss about the funding regimen or probably the sponsors, and of course the reinstantiation problem of the codes with the Galaxy codes or workflows or whatever. Any dependency problems, if at all, if we have our own node or if we have our own instance. These kind of things probably I would request Viday, to take it forward, first discussing with Nagamani and then, Kiran and Harpreet. Yeah. Thank you so much.
Thank you, Prash. Actually, I'll be regarding the educating these researchers, I'll start with that. So this is something which has been coming frequently from Narahari sir only, Shastri sir, which you are mentioning. That is actually Galaxy going to liberate the academia, the entire community from using the black box programs, and can Galaxy provide a wonderful platform for developing their own algorithms? Can researchers develop and deploy their own algorithms to solve problems with the help of Galaxy? So what will be the contribution of Galaxy in that? That's something which I'll try to put. I'd like to start with Nagamani, if you can comment on that.
Yeah, thank you for the organizing committee for giving me this opportunity. So as far as this Galaxy concerned, this from our group, especially this particular comment what Ridek has made, it is quoted by our, Sastri sir. So he's thinking about the Indian perspective about how this Galaxy will be helpful to development of open source drug discovery platform, which is very common to all. So he always used to tell like there are three issues in Indian concern, especially in India. Like most of us are using this computational, especially this computational softwares as a black box program. Just we are giving something input and we are getting some output. And also that finding some open source platform which is common to all, as rightly pointed by Dr. Parthasardis, collecting all the open source platform in a single, all the open source software in a single platform. So that is also one of the another issue. And also that most importantly, inculcate the culture of developing our own algorithms and software. And finally, the focusing on creating capacity building and large capacity building under large challenges, focusing on rather than focusing on the simple issue, which is creating the incremental outcome. So together that we can try to make a very big platform where all the people from, especially which is Indian originated, so we can try to make a very good platform. So in that case, especially as far as that workflow developments and drug design perspective, so there are multiple dependencies which we are trying to incorporate, and especially that some of the things we have developed some machine learning models which we are trying to incorporate in the Galaxy. So that time during the incorporating our own model from R or Python, so that time that we are facing little bit difficulty, but I'm not telling that that is one of the very like a hurdle to make it true, but still that we are facing. But however that it is, the Galaxy is a very user friendly to incorporate all the open source module by incorporating different chemoinformatics and bioinformatics tools by writing our own script or something. Thank you. Thank you for the opportunity.
Thank you, Nagamani. Can I request Kiran to please put some comments on this point?
Sure. Thank you, everyone. So before going directly on the point, I would just quickly say about TMS Foundation. So because it makes sense to say that. So TMS Foundation, the name basically Tarunavadanina Mukti Bharat Unyan Samstha. It basically indicates then in Sanskrit it is an open India Bharat development. So basically that's the idea of the group or the foundation. So yeah, as part of that, I'm being more of from the bioinformatics background. So this was a very core to me, or let's say to have affinity towards the Galaxy, which is I've been did my PhD in Freiburg, and having seen how the open source actually flourished here. It's like kind of a Silicon Valley, we all know, and where the Freiburg here, it's called like kind of Bio Valley. So it's like that's very much popular. So this has really a lot of scope and going in the open source. Slowly things are getting more open source, and in India as well, I hope so. So idea is as a part of TMS Foundation, we try to publish some kind of internships and then ask people to come in from various backgrounds, could be from the bioinformatics or from the computer science and others. We are very young organization. We are really going extremely as an open source, which means we don't say, please do that, please do this, only when they have time, only that the structure itself is like that, that only when they have, when they are interested. So we are going by the interest. And I'm very pleased to say, although let's say it's normal probably from the people, let's say around every time a group of 30 to 50 people will come, and then at the end of 3 to 4 were active will be found in a kind of structure what we follow. So what I'm trying to say here is this is interesting for a lot of people, but it will be much more interesting if it is Indian based, or let's say if Indian universities, or let's say Indian government organization supported universities take up this, that will be a real great motivation for most of them, that they are part of this. And the last sentence is, it's not just about only the academicians. In fact, there are many people who are from the biological natural sciences background who don't go for the jobs, and also may not be going for the researchers. They might be ending up household, so housewives or house husbands probably. So even they can actually contribute if the such a kind of platform exists. So that's the idea, to have that focus that anybody comes. So in that way we are trying to move. So yes, that's the experience with the TMS Foundation, I would say.
Wonderful, Kiran. Thank you. Can I ask Ms. Sona to please put some comments on this? Quick comments.
Yes, definitely. So the point which Dr. Kiran was telling was exactly right, because there are a lot of people who are biologists as well as bioinformaticians still having not secured a job even after PhD. So they are actually very useful resources here in this current scenario, where they can contribute so much to this, because they are not doing any other jobs. Maybe they will be doing some side jobs or something, but they can really contribute, because in order to prevent their knowledge from going down, we can actually bring them up. So that is actually a very valid point here.
Thank you, Sona. Can I ask Dr. Harpreet?
Thank you, Hriday. Hriday, I think if I summarize all the points which have been given by Kiran, Sona, and others, and Nagamani also. So as I already said, that the main point is, make people engaged in doing data intensive science. And when we make them aware of data intensive science, how they can put bioinformatics into their own biological problems, for example, and so that they can explore new dimensions of research. So they will be automatically, they will have interest in doing some using some tools. And if they can find something freely available open source like Galaxy, they will be very excited and delighted to use that. The first thing is to create awareness, which we are already doing. And I think I must mention the BioClues, because Galaxy has been launched in India mainly in collaboration with BioClues and TMS Foundation led by Kiran. So I think BioClues has already very good base, and we are trying to make people aware of that. Awareness front is already, we are being taken care, and we are accelerating every year, I think. The second important thing which is of concern to me is, and also the concern read by Professor Partha, that when we have more users, so we need a dedicated server, dedicated hardware infrastructure. That's very important. So for the time being, what we planned as a Galaxy India users, or all the active Galaxy India members, we try to uh, did a partial did a time did a very shortcut solution to this, like providing a Galaxy instance, Galaxy India instance to Galaxy Europe, and we completed the soft launch in the last year. But the main challenge still remains that we should have our own dedicated hardware or dedicated server somewhere. So I think we were looking forward to the NIST and other such government organizations where we can have funding, we can secure the funding from the government. And the second thing is what we have planned. I think that was the idea of Raj, that we can have some industry sponsor, and we can bring some satellite server, or we can also try to bring some main server at some places like for example, it was suggested to have a satellite server at HMV at my college or any other. So if we have the infrastructure in place, if we have a training plan, awareness plan in place, I think we can take the best, we can make the best use of Galaxy like platform, and we can really take our research to the next level. So I will repeat this word, that we can really take our research to the next level, which is very, very important. So if we want to match the world where world is doing what the world is doing, we have to make use of Galaxy or such open science platform on a larger scale. Thank you.
Yeah, so if I can just interrupt here, Viday. Yeah, I think this has really gone to a wonderful thing. We have certainly discussed about the funding regimen and the sponsorship and all, wonderfully tabulated by all the speakers. So echoing all these sentiments, I think, on a lighter note, we have hijacked Kiran from TMS Foundation into BioClose. So now we have a so-called, more capacity building. And as Nagamani and CSR Nees from Narayana sir have wonderfully pointed out that we have these servers in place now. So if this could be started, or even otherwise, if this need not be started, we have already found out some kind of sponsors who could pay anywhere between 30 to 40,000 rupees per month. So that would be enough to run a kind of a steady end server for at least 2,000 users every day, from an Indian point of view. So that can be taken up on a kind of a cloud-based structure entity where the sponsorship amount can be leveraged into that, or we can have a kind of a physical server like Harpreet, who has pointed out, that can be put up in CSR East with Hansraj Mahila Vidyalaya as a node. I think I would like to request the hosts as well as the panelists here to kindly think about this angle as well. This will be a wonderful kickstart for development of Galaxy instance in India. Yeah. So yeah, over to you, Viday. Yeah, sorry, Kiran. Yeah.
On that point of hijacking, I wouldn't say it's a hijacking because the TMS Foundation itself is the concept like that, that because it's not just in one field, it's an open for many fields. So it's not about going in the bioinformatics. It's also right now in the tax and finance related things. So there are a couple of other branches also there. So it's basically clubbing with other such organizations and then do. So for the bioinformatics, nothing other than the BioClose I can see right now in India, where I can club in.
That was certainly on a lighter note, Kiran, when I said hijacking. Yeah, thank you.
No,
so
I was also in the light. Yeah, sure.
We all need to come together for sure. Yeah, Viday, please. Yeah, please go ahead.
Yeah, thank you, Brash. Thank you, Kiran. We have Gareth with us, I think. So I think it's the right time to ask about the dependency problems which comes generally when we're trying to host a server in a particular location physically. So if Gareth can put some light on that. So if we're trying to start a new, like Galaxy India or something, so what are the various problems or what are the various requirements which will be there to facilitate everything? We'll start with Gareth and then move ahead.
Thank you so much. So yes, the context is Galaxy Australia runs all its infrastructure nationally, but not on a single head node. We have made significant use of Galaxy's Pulsar network. So the challenges that have come with that are a few. So the first primary challenge was sufficiently resourcing the head node for multiple users. So this actually has nothing to do with the computational capacity, the number of worker nodes. This was around simply building a robust and redundant web service so people get a responsive interaction with Galaxy. We actually have outgrown our head node a few times in our 4-year journey, and we've picked up and moved around the country, and that is not a small process. Moving Galaxy is actually moving your entire user database of data, and that's in the many, many terabytes of data that you have to rsync or take offline and prevent people doing analysis during that process. So we don't take that movement lightly at all. one of the more successful policies we have put in place, which we really did value and still value, is we have put a temporal time frame on people's data. So we have defined Galaxy Australia very strictly as an analysis platform and not a storage platform. So any history that someone does not access for 365 days, any complete history, not individual data set, not popping back in to check something, but something, a complete Galaxy history that's sat untouched for a year, we will give users a 2-week notification period and then purge it from the system. So we actually now know from a responsible management point of view of our service that we're actually 90% of the data we hold is current to the last year. There are a number of legacy projects and there are a number of long-running projects, but this allows us to engage with our infrastructure providers and give them really clear definitions and mandates about the amount of storage we need, the redundancy in storage, and the expected growth rates in storage per month and per year. So for us, that's been a really positive management move for the service. I'll then take 2 or 3 more minutes. I guess the next level engagement comes with those members of the Australian community, in our case, that want to bring a Pulsar service to our environment. So for them, this has included our supercompute centres and some of our largest university institutes, and they get, quite frankly, they get the accolades of help running a successful service. So they get their partner logo on Galaxy Australia, they get accreditation in talks. We can, for every one of those partners, we can give a robust report of how much their service is used, the kind of users that are using it, the kind of people from the own institute that are accessing it. So there's an upsell value there. Most tools work on the Pulsar network, but not all of them. But it has allowed us to engage with more partners to go down that route. Instead of looking for that one massive pot of computation, we're looking for smaller pots of computation all around the country, which together add up much larger than we can host at our head node. The final nuance on that is at least one of our providers of Pulsar, and we're grateful for all of them, but one of us is our national computational infrastructure in Canberra, and their infrastructure has quite high data security access requirements. So we have actually entered into quite a formal relationship with them. We have committed to certain styles of users and jobs being run on their infrastructure so that they can conform to their ongoing data policy needs. So each of those engagements with your Pulsar can be quite a specific interaction you have, but a positive one. And I think I'll leave it there. And if there's questions, that's great.
Wonderful, Gareth. Wonderfully inserted. Thank you so much. Yeah. So I'll probably move to the last point of it, something on the sidelines of what Vijay also mentioned, or probably others have mentioned. In fact, what I probably propose here is, whenever we talk about workflow developments, we in India, as we discussed, very often that over 70% of the users are chemists, and they practice chemoinformatics or rational drug design. So I was just wondering, why not, the Women in Biology Forum, where, Sona Charles, can probably educate the women, the households, or the women, who are sitting at home. They can probably, come up with these workflow prototypes and develop some workflow prototypes and come up with, some kind of, success stories. This is where, we in India at this point of time, we really need it. We have, probably educated about 800 researchers and 800 students, and introduced probably Galaxy to them. But in order to ensure that our our instance survives for the next 2 or 3 years, we need more capacity building, and I believe that this makes absolute sense when we can come together. So do you have any quick say, maybe something to chip on, maybe Harpreet or Sona Charles or Nagamani?
Yes, Pras, I think it's really a very nice suggestion. And as already told, that the training and awareness is very much required at this time. And if we can use the resources like the women which are having some time at household, and even we can target some of the the women folk working in research institutes or in the colleges, and they also want to do research, but they do not have resources. So I think if we can train them, if we can have a consortium, we can have build a groups, build some research groups. We can target them first using the already available workflows, and slowly we can train them how to develop their own workflows. And as you said that the drug discovery regime in India is very popular, and many of the people are aware of these things. So we can make them aware of how they can try to make best use of Galaxy, like open source platforms, and then or to make best use of their ideal time, and also they can progress in their career. So it's not only learning something, but they will be highly benefited from such research papers, such publications, such projects, and they can even apply. They can develop a confidence to apply some very good grants, and I think that can lead to the development of women in biology or women in science as a whole. That's a nice idea, Prash. Thank you.
Yeah, thank you, Harpreet. Sona, do you have any quick say, please? 30 seconds max. Yeah.
Yes. Actually, along with what Harpreet sir has told, I would suggest taking up students who are wishing to have internships also in this regard, because during this COVID scenario, what I have observed is most of them are not able to move from their parent institutions to any other institution for taking up their projects for their as a part of their MSc or curriculum. So the most part is we can utilize them also in this case, because they would be requiring an online platform which can be commonly accessed by both the mentor as well as the students as well. So like that, we can just create a kind of mutual understanding about the Galaxy platform. We will tutor them as well as we'll get inputs from them also for developing Galaxy. So I think that will also work in one way.
Thank you, Sona. So to some of you who don't know, in fact, we have known Sona Chalice for over a decade, I believe, the last 10 years. She has grown as a wonderful scientist right from her early academic days. Now she's a group leader. She mentors her own students. She has been an active practitioner of or user of Galaxy, and the kind of inspiration one could garner from people like Sona Charles, that can really be very much motivating. There are many young students over here, especially the women researchers. I think Galaxy is one such kind of a platform where you can take leaps and bounds with your research sitting at home. Yeah, thank you so much, Sona. So Kiran and Nagamani, do you have any one quick say before I go to the final point? Yeah, thank you.
I just want to add only one point. Even if the student, they would, if they are very much interested, if they are developing some source code or something, even we will be very happy to deploy those code in the Galaxy, even though that we can publish in our Galaxy server, and the students also will be getting more motivated. That's it. Thank you.
Thank you, Nagamani. Yeah, Kiran. Yeah.
I think there are two things here. One is using Galaxy itself, which is already a popularly used when the Galaxy instances are already available from other countries, or let's say from the Australian, European, and this thing. This they can already start. Another thing is within India, that is the point is more about how do let's say which institute or which organization or which entity can manage that resources. Let's say if some people are coming, so many housewives or whatever, what is the place of source? That is more a kind of, if it gets more organized, that will be improving more. So I'm sure this is going with the Galaxy India, it's going in good direction. Like at least in the Galaxy project.in, we are trying to put everything over there. So that's the point I want to take at this point.
Right. Thank you, Kiran. So we have the last 4 minutes, unfortunately, but this is a very important problem that I wanted to discuss with Gareth and probably Kiran. We thought that there are, of course, now several instances, several continental Galaxy nodes or instances or servers. There has been a kind of a problem, with not only with duplication of the users or probably, deduplication of the users, but also, duplication with respect to the number of workflows that have been promulgated, the number of workflows that people have been using over the years. And at some point of time, in order to save not only the active space, but also the context, we might want to have some kind of blockades, in ensuring that the users do not have multiple accounts across several servers or maybe within the same server, and also something on the sidelines of ensuring or keeping the spirits high, so that the same workflow could be rather better shaped, rather reusing the same kind of a workflow for a number of users. So this is the point that I wanted to make it. Maybe these are two different points. Gareth, do you have any quick say on this, please? Yeah, thank you.
Yes, I do. I'll try to keep it quick. Multiple users on multiple galaxies, really interesting. Personally, I have accounts on multiple galaxies, and I do that as part of my management. So, whoops. That said, there is aspirations for a global sign-in, so that once you sign into one galaxy, you're signed into all of them. In terms of workflows, fully agree. We need a clearer route to identify best practice workflows that by popular demand or learned individual citation are brought to the top of the list for routine execution. As a slightly tangential statement, but a quick one, I think Galaxy Australia struggles with how do we scale as a team of service engineers, essentially, to an entire community. And we're really thrilled that the latest version of Galaxy that's been out for about 2 weeks has a new customizable landing page for new users. So this is someone who arrives at the service the first time. You get a completely different look and feel to Galaxy with an opportunity to educate them on best practice in Galaxy, the clearest resources for them, a how-to guide, whatever you really want to put up there. And to me, these are the solutions that we need to put in place so that we scale beyond a small number of individuals trying to engage with a large number, and we make that engagement more streamlined.
Perfect. Well said. Well said. Thank you. Thank you so much, Gareth. So, Kiran, 20 seconds, please. Yeah.
Yes, that's a really interesting point. I think multiple galaxies, multiple accounts in multiple galaxies is just that it shows how much interest is there. I would take it in a positive way. Of course, we still say that not in the same Galaxy instance, not to have multiple accounts, that at least hopefully that not people are doing. Yes, the only thing is if we can gather, or let's say can organize the resources what we spoke just now, like household wives or other resources and other things, because it's a huge number. It's if it start taking, if we have a better organizing thing, then I think we can do a lot.
Yeah, thank you. Nagamani and Hriday, probably, since if we are to probably set up this instance or server in CSIR NIST, how we can probably solve this duplication problem? Do you have any quick say for 30 seconds? Yeah.
No, that I just have to see that once we have set up the server, then we have to think about, whether we can put any, like, whether we can put any filter or something. We have to think about it. So still that we didn't think about this, to how to solve the duplication if multiple user or if the same user with the multiple account, if he is trying to submit a calculation or something like that.
Yeah, thank you. Thank you. Harpreet, one final point on your entity on duplication of workflows. Do you have any quick say? 20 seconds, please. Yeah.
Yeah, thank you, Prash. I think I agree with the points given by Gareth and Kiran. Duplicacy has positive and negative impact. I think we will be happy to have duplicates, more users in the initial phase, but when we go forward, we have to devise a strategy to remove that duplication and make more sense. That's the only thing.
Yeah. So thank you so much, dear panelists. I'll just quickly wrap up what we did discuss over the last probably 45 minutes. This is a kind of a curtailed panel discussion, but we learned some wonderful lessons today. Thank you so much, elite speakers. In fact, we started with Sona Charles and Kiran and Harpreet and Nagamani, who gave a wonderful gist about the kind of challenges that we have been facing in educating the masses, in educating the researchers, take up the galaxy, and the kind of the workflow problems, the kind of motivation that they need to sitting at homes during the post-COVID times and other stuff. It's not only the capacity building. We also have had Dr. Pardasardi, who spoke a wonderfully giving a kind of a gist about galaxy towards the toxicology usage, the toxicology research that he has been doing over the years, and how his students have really been trained and mentored and have given some kind of impetus towards the capacity building. And we have, of course, Harpreet then chipped in saying that it's not just the capacity building, it's also this so-called the trust building. We need to inculcate that kind of trust among the student brethren. Of course, there are some kind of problems, but these problems could be converted into some kind of wonderful solutions wherein we encourage the younger brethren towards this. And of course, the Women in Biology Forum from BioClues, where we have a good strength of almost 1,600 women. They can be trained because they could sit at homes and do wonderful workflow management towards usage of Galaxy. And of course, Nagamani and Hriday has taken a wonderful point on how CSR NIST can be used as a kind of a node or development of a server hub over there with dedicated hardware and software that could be promulgated in the future. And Gareth has wonderful points, two wonderful points, one of which I really liked that I would like to mention, that it's not Galaxy should not be taken as a kind of a storage platform, but as a kind of an analysis platform, which everyone has to realize. It's not that we ensure that we dig our histories, we probably we use those particular things only for our management of workflows, but also towards development of best practices, so that we'll not duplicate or come up with the so-called redundant workflows again and again. So these were some of the points I think we all discussed today. I think there is certainly kind of a need of the hour to ensure that the Galaxy instance would be kept alive. But Gareth has given that wonderful life during the workshop, where we had that kind of instance, where he has shown from our Europe node, India dot. That has really put us on the map, I would say. A couple of years ago, we were just dreaming about Galaxy India. Last year, we dreamt and we kept it alive. And today, we are being alive, and we will be living for generations where the young students can bring this Galaxy India to the fore. I think we should really thank all the elite panelists for spending this wonderful time and energy today. So let's give a big round of applause to all the speakers. And not to forget the big take, the photograph. So please, yes, I think I would request Sugu to help us with this photograph. I don't know how to make it in one screen. Yeah, please. Yeah. So all smiling faces to be on, please. Yeah, thank you. Sorry, is someone taking a photograph?
Yeah. Let's smile.
So, yeah, Pardasardi, please remind switching on your camera. Yeah, thank you.
All the participants, can you please?
Participants as well, please.
Yeah.
Okay.
So thank you and all. I think.
Thank you very much.
Yeah, we have the keynote now. We are just 3 minutes delayed. Yeah, thank you so much. Yeah, over to you all.
Hi, Alberto. Hello, Professor Alberto. Oh, Enzo. Hello. Yeah.
Sir, you have to unmute yourself, sir.
Dr. Nagamani, whenever ready, he's ready, we can start the session. Yeah.
Yes, sir. We can start.
Can you see my screen?
Yes, perfect.
Okay. Then I shall begin.
Yeah, just wait for a confirmation from the organizers.
Oh, okay, sure. I'm just making sure you can see everything.
Yep.
Can you confirm me how long I have?
No, I think you have plenty of time. And
no, I know.
It's
actually 25 minutes.
45 minutes.
Yeah, it is.
No. It's 45 minutes plus 15 minutes Q&A. Yeah.
Yeah, 45 minutes plus 15 minutes.
Yeah. It's a keynote. Yeah. Thank you.
Yeah, an hour. So if it's okay, we can start now.
Yes.
Okay. So once again, very good afternoon, everyone, and it's happy to chair this session and thank the organizers for this opportunity. And with a follow-up with our previous discussion or panel discussion, is the kind of a very off topic where Professor Alfredo Benso is going to talk about. I welcome him for our InBix conference. And before starting, I'd like to give him a brief introduction of Professor Alberto Benso. Is an associate professor in computer engineering at Politecnico di Torino, Italy, where he teaches operating systems, hypermedia design, and fundamentals of computer science. He has a PhD in computer engineering, and since 2018, he is also an adjunct professor of electrical and computer engineering at the University of Illinois at Chicago. He had teaching and research position at University of California, San Diego, and University of Texas, Austin. It's great, very impressive. And his research interest and focus are on system and computational biology. He scientifically have a lot of collaboration with several universities and notable industries on worldwide. He has involved in several computer society and sponsored conferences and association with various membership of various eminent committees. And Benso also senior member of IEEE. And he received several awards and outstanding contribution awards, a continuing service awards, and Golden Core Award. And since 2003, he has also been volunteering several CS committees and conferences, and is very, very active. And his scientifically, his publications are outstanding. He has published more than 150 research articles in journals of high impact. And with that, I invite him to deliver his talk on surviving the data era. hopefully we survived on the COVID-19. So now the data era is going to start now. Yeah, thank you, Professor. It's over to you.
Thank you. Thank you very much. So thank you very much for inviting me for this presentation. I I decided to title this talk Surviving the Data Era because we are in a moment where apparently data is controlling every news and every aspect of our society, and obviously we work in a topic that is very data dependent. And so I wanted to just point out some of the criticalities of how we as life science researchers have to handle data. It seems something very basic and very trivial, but it's actually a critical point that could affect the whole process of our research. So just a few words about me. The chair already said more than enough. What I want to point out here is that I was not a life science researcher from the beginning. I started my research activity as an engineer and working on hardware and software reliability. And then about 15 years ago, I moved to biology and systems biology. But this gave me also the opportunity of approaching this very interesting topic, beautiful topic, with engineering mind, an engineering point of view. And I think that this is a very important aspect of how to approach the world of life sciences, because the engineering world and the life science world don't share much in methodology, research methodology, in computer expertise, and it is necessary to grow students and researchers that have both state of minds, the engineering side and the life science side. And I will try to discuss this focusing on the data in this presentation. So the basic outline of the presentation is just talking about system biology and data science and how the two relate to each other. And to do this, I would like to start from the very beginning and trying to give you a definition of bioinformatics and systems biology from an engineering point of view. So systems biology is the computational and mathematical modeling of very complex biological systems. So the first questions I want to ask you is, so do you know what is a system? And what is particular about biological systems? So I found this very simple definition that says that a system is a collection of parts and factors that work together to complete a task. And for me, it's extremely important to focus on the first part of this definition, a collection of parts that work together. This is very important because this definition forces us to concentrate to the fact that we are studying a system that is made of many subsystems, of many parts that work together. Now, the second very important point is that what do we do with the system? We're not classical engineers that build a system, but we are instead researchers that have the system already made, and we're trying to understand how it works. And that is called reverse engineering. And reverse engineering needs to be approached with very strict scientific methodologies, but the way you approach the problem depends on the type of system you are studying. And biological systems are complex systems. They are not linear systems, they are complex systems. What is a complex system? A complex system is a system, is a system which, as we saw from the definition before, is defined by the parts that made up the system. But the very important characteristic of complex systems is that some functionalities and some properties of the systems emerge from the interaction of their parts. This is the most important things of all. If some properties emerge from the interaction of their parts, the consequence is that these properties cannot be predicted just by studying each part individually. We need to study the whole system working together, okay? Just as a very simple example, LP player, music player, is made of a lot of very small interacting components, mechanical components, which produce music. And if you study each individual part of a record player, you will never be able to understand or figure out what music is. You have to play the whole system together to generate the music. And the same thing happens with life and with our consciousness, no? With the way our biological system work. What you have at the end, what the biological system is able to do, is not possible to understand if we just study the individual proteins, the individual cells, the individual organs. We have to study the whole thing together. And the other thing is that the complex system dynamics, or the way they behave, heavily depend on the initial condition and perturbation. This makes the gathering of the data extremely difficult. So how do we approach this challenge of understanding how biological systems work? We use systems biology. Systems biology is, let's say, a mix between the top-down and bottom-up approaches that are taught in engineering as the two main approaches for reverse engineering a system, so to understand how a system works. Systems biology is a mix of the two, and it's based on this very nice circular pipeline. We start with lab experiments together to stimulate the system we are studying, and to gather data about it. Okay, then from all this huge amount of data, and now the technology allows us to produce enormous amount of data, we create models. So we try to model the system in a simpler way, to try to recreate a mock system that behaves, obviously with limitation, with simplifications, the same way that the real system works. From this model, we simulate, so we try to make it behave in some way, applying stimulus and reading his output, and we try to understand how it is made inside. So what is that allows the system to work in that way. So, and from there, we make new hypotheses that lead us to new wet lab experiments, to new data collection, to refining the modeling, to more simulations, and so on and so on. And we keep going through this loop. And at each iteration of this loop, we refine and we increase the quality of the model. Now here, the weakest point is the data collection. If we are not able to treat and guarantee the quality of the data that we collect from the lab experiment, we waste time in the experiments, we will never obtain a reliable model, and so this part of the pipeline can affect the whole process. Okay, so in this talk I'm concentrating on that particular aspect of the systems biology pipeline, the data collection and data handling in general. Okay, data is so important right now also because of the evolution in artificial intelligence that data can be considered almost as the new oil. And the artificial intelligence is the new electricity, which is permitted thanks to this new huge amount of fuel, which is the data. But you know, the process that we are trying to do with the data in general, but here in particular, in the life science world in particular, is that we want to extract knowledge from the data. Okay, and in this process we have to pass through several steps. First, you have the data, which is the raw material. Then you have the information. The information is contextualized data. Okay, so it's the data considered in a certain system, in a certain environment, under certain environmental conditions. From this information, sometimes, if you're good and you have enough resources, you can extract knowledge, which is a useful information that is somewhere captured in the information concerning a specific goal. Okay, and then there is a fourth stage, which is the wisdom, which is something that we can create behind the knowledge, and which is the step that the artificial intelligence is not able, or maybe will never be able to reach. The artificial intelligence will always stop at the knowledge stage. Okay, so given this transformation of the data into knowledge and wisdom, we need to acknowledge the fact that the path from this data to the knowledge is not always straightforward and easy, and especially that there could be no information or knowledge in the data. So there is no guarantee that in the data that we collect, there is always something interesting and something useful to extract. And that sometimes to actually extract good information, you need to require more experiments. For example, if you have a lot of data about gene expression, you know that the statistical correlation between two genes does not mean causality. Does not mean that one gene is actually controlling, regulating the other. And to discover this, you need further experiments and further modeling. So this process of extracting the data is even better represented by the following slide. You have the data, okay? Some of it is interpretable, and some of it is uninterpretable. Okay, think about some really big data repository, for example, from next generation sequencing or some proteomics or genomics, I don't know, some microarray data. Not all of it is always understandable. Then in the interpretable part of data, you have part that is relevant to the problem and some that is totally irrelevant to the problem that you are considering. Okay, and this is still data, just data. So in this huge data universe, you have a part of this data which is information. And information we can here define it as non-redundant part of the data. So what is useful of this data is actually useful means relevant and interpretable. Okay, now let's focus just on the information part. With this information that you selected from the data, okay, part of it is still not interpretable and part of it is still interpretable. You're obviously interested in the interpretable part. You have noise, which is info, but is irrelevant. And then you have your signal, which is the relevant information. But from the signal, from the relevant information, you have to identify the insights, so the new information, which might be a little portion of that signal. So you see that from the huge amount of data, you just need this ocean of data. We are just trying to understand, to find a drop of water, which is the really new information. And to have any hope to select and to find this information, you need to have some scientific way of handling the data. Otherwise, you'll never be able to find that new information, okay? Otherwise, just collecting data is not enough. If we don't have structure data, integratable data, then all the stuff that we collect is wrong, because we try to do precision work, precision guesswork, because we're guessing how the system is working. We don't know. We're just making hypotheses based on totally unreliable data. So we want to avoid, okay, to try to do something really precise and figure out incredibly good algorithms if then we apply them on data that is not reliable and it has a low level of quality. And we need to try to define what this quality is. So the first point that I want to make is that to extract information requires rigorous scientific methods, okay? Information extractions requires rigorous scientific methods. Which is not done by the media, for example, okay, which is very good at taking data and reshaping it, or trying to extract the information they want to extract, not the information that is in the data. They first decide the information they want to extract, and then they try to look at the data so that they can claim that the data says something that it doesn't say. And if you are interested in this type of thinking, I suggest you to read this book, A Field Guide to Lies and Statistics, which is a very, very interesting book, pointing out the typical ways data is misinterpreted and misused, okay? Now, I hope I convinced you that the data is very important in our topic in life science and in systems biology, that collecting quality data is a key point in hoping to generate some new knowledge. And now I want to focus in the main challenges of this data revolution, okay? Main challenges that are the size, the heterogeneity of data, the falsification of data, and the quality. And I will mostly focus on the quality, but I also want to mention the other three problems, because maybe they're not all clear to everybody. I will be very quick on the first, on the size issue. We know that technology now allows us to generate a huge quantities of data. The cost of sequencing the DNA has fallen over 100,000 times in the last 10 years. Now it's extremely cheap to sequence DNA, even it's now possible to sequence single cells. We are starting to approach the study of the genome, for example, and the microbiome. And if you think about it, the microbiome has from 200 to 2,000 as many DNA genes as our human cells. And we still have problems understanding human cells, but we want to approach the study of something that is 200, 2,000 times more complex and interacts very heavily with our physiology. Okay, so the amount of data that we have is so big that if we don't understand the implication of not handling a good data, then we have big problems. So size is one problem. Second problem is heterogeneity of the data, okay? For many of you that worked with real data, you pretty soon discover that it's very difficult to make data taken from different sources work together, okay? We have a lot of online databases which are not easily integratable. It's not always easy to take one database and create relationships between the records of another database. Medical data sometimes is the worst because it is taken and archived by people that are great scientists, great doctors, but have no knowledge of the problems of creating data, how you create the data, and which mistakes you should not do when you collect data from patients, from machines, from lab exams. And then you have a lot of proprietary data, which is kept like a secret and not shared with anybody, and on which a lot of studies are based that become very difficult to replicate and to validate. Okay. Moreover, in our field of life science, there is another problem which is not so common in other fields, and it's the problem of the standards. What happens is that in many times bioinformaticians are not happy with the available standards, and so on their point of view, solve the problem, they create a new standard. So the situation was, in this comic, there were 14 competing standards, and we didn't like the 14 competing standards, so we create a new one. So now we have 15 competing standards. And this is a problem, because if every data set follows a different standard, then it becomes extremely difficult to have different standards work together, okay? And we must have the ability of integrating data. We must have the ability to integrate data, because otherwise we will never be able to extract information. Hearing a lot of noise. Is there any problem?
Yes, we will look into that. Just okay, please go on. Yeah.
So as I was saying before, since we cannot limit ourselves on studying the isolated parts of the system, but we have to study the whole thing together, we must be able to integrate data and take data that comes from different aspects of the system, from different subcomponents of the system, and analyze it together. Because since this we are studying a complex system, its properties emerge from the interaction, from the parts working together. And we have to see the parts working together to understand the system. Another problem is falsification. I was really surprised when I learned about this from a colleague and friend of mine in Italy that did a lot of research on falsification. And he discovered that in an incredible number of papers published by important journals, there were experiments that were never performed with fake data published, or the experiment was performed, but then the data was altered and changed to make it fit the claims of the authors, or even the experiment was performed, but then the data was duplicated and used for different purposes. And this can lead to fabrication, so just come up with results that you never produced, falsification, so just change the results, or plagiarism, which means reusing results from others and attribute them to your own. And to make you understand the size of the problem, in this test, or this sample of submitted manuscript from June to August 2017 to 5 independent journals, 20% of the papers had manipulated images. Manipulated images means that either they were copied by other papers or they were edited with Photoshop. So the importance of a rigorous review of the articles that we are asked to review is very important. I know it takes a lot of time, but if we don't contribute to find these problems and to identify these falsifications, we actually contribute in putting out data and informations that are wrong and that can affect a lot of scientific efforts. So apart from this, which obviously is voluntary falsification of the data, what I want to focus now is the fourth item, which was data quality. Okay. So data quality, and obviously what dirty data means. We need to define what data quality is. And to do that, we first start from a definition of data science. Data science is a, let me say, new, even though it's not very new, but new discipline that is in the middle of computer science, math and statistics, and domain knowledge. What's domain knowledge? It means that a data scientist that analyzes data cannot only be a computer science genius and a statistical or math genius. He also needs to know very well the domain in which he's working. Okay. Again, for the same reason that I was telling you before, because a data scientist studies complex systems, and complex systems means that they are systems made of parts that work together, and the properties emerge from this, from the working together of the parts. So if you don't know the domain you're analyzing the data, it's very unlikely that you will understand anything from the data, and you will be able to make hypotheses on something that you don't know as much as possible. Okay, so given this definition of data science made of three big components: computer science, math and statistics, and the domain knowledge, what is the definition of dirty data? So from the statistical point of view, so from the mathematician point of view, it says, oh, look, there is a process that produces data, and any data set is a sample of the output of that process. The results are probabilistic, And you can correct bias in your sample, for example, by applying statistical methods. So the statistician relies on his mathematical modeling mind to correct and understand the data. The computer science guy, he has a database view in his hand. He has an entity relationship view in his hand. He understands that some of the in the database are missing, are corrupted, are wrong, are duplicated, but he relies on the relational model that upon which the database has been constructed. And so just enforcing some relationship rules between the data, he hopes to be able to improve the quality of the value in the data set and discard what is not working. The domain expert, instead, which could be the biologist, for example, he doesn't understand all the database stuff, the mathematical stuff, but he looks at the data and they don't look right. And the answer that they suggest doesn't look right. And so he asks himself, what happened? But he cannot answer because he doesn't have all the other expertise. So the data scientist, instead, view of dirty data is some combination of all the above. The data scientist has the ability to understand why a data is dirty from 3 different points of view, okay? And that is critical in his work. So now, why then data can be affected? Quality of the data can be affected because data is dirty on its own. It means that data has been collected in the wrong way and it's noisy, it's not well structured, for example. Or you start from very clean data sets, but their integration screws them up completely. Data sets are clean, but suffer bit rot. Can data expire, for example? I would say yes. Imagine, for example, a lot of the releases on the microRNA data, okay? From one release to the other, there are a lot of changes, not only because there is new information that is added to these databases, but also because they changed names. I mean, they decided to change the names of some microRNA from one release to the next, so that if depending on which release of the Myrna database you're using, the same name corresponds to 2 different sequences. This is crazy, but still it's what happens. And that means that you need to link the data that you're using to its release, to its time frame, because sometimes you cannot mix 2 data sets that come from different time periods because the meaning of that data is different. Or obviously any combination of the above, okay? Just to give you a very quick, dirty example, if I get something like that, it's like the first 2 lines of these slides of data, okay? A file with a lot of lines, all like this, which is the way we receive data from our colleagues, from our friends, from the doctors when we have to analyze data, okay? We just receive a file with a lot of information. Do we receive a file that explains what each field means? What is the key? If each field is unique or not? The unit of measure of each field, okay? And sometimes these files have typos, have data in multiple formats, have missing or default values. Sometimes, for example, in biology, life scientists or doctors don't understand the difference between a missing value or 0 value. So if they don't have a value, they put 0, and that is super wrong. There are these little minor trivial mistakes that make the data dirty, and that are very difficult to prevent too. For example, in this data, if I tell you that the field number 3, this 24.95 and 2000 is revenue, is it what? Revenue in dollars, in cents, in euros? Which is the how do I quantify that number? Field 7 is usage. The last one. Why is it 1,000? What does it mean? And so on. And sometimes we receive the explanation of the data in an email from our colleagues, which says, oh yeah, you know, the first column is this, the second column is that, and then you look at the columns and it doesn't look like it's like he said on the email. So you go back and forth trying to understand what it means. There isn't a document. There isn't the habit of creating a minimum of annotation to the data. And then we have data glitches, so systemic changes to data which are external to the recorded process. Okay, so for example, the data layout changes. You pass from one Excel to the other, for example, American Excel and Italian Excel, and the comma becomes points and the points become comma, or some fields swap position in the formulas, or the system consider a value dollar versus euros because it's something automatic that the software is doing, or the software treats in different way missing and default values, or, and this is very, very common, there are gaps in time series. You are being asked to analyze time series and sometimes a value is missing, and they say like, yeah, we don't have that value. And what do? You just put a number, a random number. There are techniques to fill that gap, but you need to know the phenomenon, the system that you're studying to correctly fill a gap in a time series, for example. These are other dirty data problems that I took from a Stanford course. Problem with separators. With naming conventions. For example, cities called, same city calls in different ways, same microRNA called with different names. Different representations of the same data, 2 versus 2 written in letters, or fields that are too long, violation of primary keys, not understanding of primary keys, of what a primary key is, which is a number or an information that uniquely identifies one record. Okay, so like your student ID is your primary key in when you did university, or your social security number is your primary key as a citizen. And this concept of primary key is completely lost in hundreds of data sets. Redundant records, so information that is redundant and that is not updated correctly. So you have redundant records which mismatch. And then you have all the licensing and privacy issues that are real or sometimes just used to prevent data from being shared among researchers. In my opinion, data should be public and should be shareable with everybody without any limitation, except maybe anonymity. So is data cleaning what will make everything okay? Assuming that we have a way of cleaning the data, would it make everything okay? Probably not. Let me tell you this story. The appearance of a hole in the Earth's ozone layer over Antarctica was first detected in 1976. But it was so unexpected that scientists didn't pay attention to what their instruments were telling them. They thought their instruments were malfunctioning. the data were rejected as unreasonable by data quality control algorithms. Okay, so is the data quality control algorithm, even though at that times maybe was not as sophisticated as it could be now, that rejected the data as unreasonable. So too much data cleaning based on algorithm can be a problem because can make you lose details. Okay, but data cleaning is a problem. Look at this. The cost of dirty data exceeds $600 billion for US businesses annually. Okay, $600 billion just because the data is not clean. So at this point of the presentation, we are over 39 minutes, so I need to go quicker now at the end. Let me just show you a few slides about data quality. What is conventional data quality? Is accuracy, how the data was recorded, completeness, if all the data was recorded, uniqueness, if every data was recorded once and there is no duplication of data, and timeliness, how the data is kept up to date, and consistency, if the data agrees with itself, there are no data that fight to each other. Okay, and the problem with this definition is that this definition is unmeasurable, is context independent, is incomplete, and vague. Okay, because we have many different types of data, We have federated data, high dimensional data, descriptive data, longitudinal data, streaming data. We have the data has a shape. It's not all the same. Each data is different from another in the distribution, in the way it is describing the system from which it was gathered. Okay, and there are many different uses of data. So quality also has to reflect the type and the use that you make of the data. You need to know the rules behind the data to define quality. So we need a definition of data quality that reflects the use of the data and leads to improvement in the process and is measurable. Because if we cannot measure it, if it's just a quality description, then it's very difficult to use it as a measure of your process. Okay, so to do this, we need to understand what is the data quality continuum. What does it mean? Which is the history of a data from when it is born, gathered, to when it is analyzed, and understand where are the weak points. Okay, so the data continuum is this. There's the gathering, the delivering, the storage, the integration with other data, retrieval when we need to analyze the data, and the analysis itself. And in each of these items, we have some big problems and possible solutions. I had them here in the slide. You can look at them later. I don't want to overuse my time, so I will go to the last line. I will provide this presentation to the organizers so they can share it with you if you want to go more in details of this last aspect. Let me just conclude that saying that this data quality continuum has to be monitored, and the only way to correctly approach this problem is a multidisciplinary approach, because no approach will solve all problems. We need process management, so we need to create processes for data quality. Okay, we need statistics that focus on the analysis and try to understand where the result is correct or not. and try to find and repair, because we can repair some dirty data with statistics. It's important to know database theory to focus on relationship and ensure the consistency of the data, of the key, the primary keys, the indexes, the relationships and stuff. And then we need the metadata. So we need the domain expertise, the ability to interpret the data and understand what it means. And sorry that I ran a little long, but I thought that were interesting items to talk about. Thank you very much.
Thank you. No, that was completely fine. Thank you very much. So I would ask the audience to have put their questions into the chat. And there is already one question from there, which I would ask you, which is, when gene expression data doesn't correlate statistically, how to fix this problem?
Well, you need more more experiments. Obviously, gene expression data doesn't correlate statistically. It could mean that there is no causality between the two genes, so one is not expressing the other. Or we need to remember that when we, or we cannot say, because as you know, when we do gene expression data, we do an average expression. We don't actually monitor the dynamic of the expression. We do an average of the expression. So we will never know without figuring out a more precise experiment that investigate that particular relationship.
Okay.
How to take care of duplicate names data from different resources? We, for example, we created a database called the Ring database, where we try to integrate 36 or 39 different databases concerning regulation. So gene regulation, protein to protein interaction, drug regulation, drug interactions and stuff like that. And you have to do it by hand. If there is no technical description of the data, if nobody follows available methodologies to describe the data, and then you have to do it by hand, unfortunately. And this means also losing a lot of data and therefore information, because for sure you will make mistakes doing this. If you have to do it by hand, you'll make mistakes every time.
Okay, I have a question. Understandably, metagenomics data is a huge compared to the genome sequencing itself. How do you use the data as of now? Like, is it a challenging to address the huge data problem, or is it advantage that we have metagenomics data?
It is, well, it is both, because it's an advantage, because the more data you have, the more research space you have. So the more probability you have to find something useful and new. But obviously the reverse side is that you need methodologies that are able to discriminate between, what I showed you before, what is relevant, what is irrelevant, what is the signal, and what is the noise. And so I think it's an opportunity, and I think that a lot of people is taking advantage of that opportunity for good research, and a lot of people is taking advantage of that opportunity to feed bad information to people. So yeah, it's a very critical situation, ours.
And the last question, so like dirty data costing around 600 billion, the statistics which you gave, I presume that's a kind of a decade-old stat, I suppose, because the image has 2012 ISON. Did it increase more in the last decade, or were able to address?
I didn't find the very reliable information about more
Okay.
Recent.
Okay.
But yes, it's absolute. Imagine that the more data you have, the more dirt you have. So
yes.
you have more cleaning to do, and it's not only the cost. That was not only the cost of cleaning the data. It was the cost, maybe the lost revenue because of dirty data. So the more data you have, for sure, the more problems and issues you have.
Okay, I hope as ozone player we are not ignoring that. So on that note, I will end this session. Thank you, Professor Alfredo.
Thank you.
Yeah.
Very much to everybody.
Yeah.
Yeah.
Sugrakar,
a quick photograph, please.
Yeah, sure.
I request all the participants to please. Yeah, please. Only 5 in the audience. There are a lot of audience, right? Yeah, please. Could you please turn on your camera? Yeah, please, everyone. I want more crowd to be there. That's fine. Yeah, thank you so much. Yeah, thank you one and all. Thank you.
Thank you, Dr. Sugnaka. So it's up to you. Over to you.
Now I think we have a
Thank
quick
you, sir.
time.
Yeah.
It's 1.
Yeah, 1 to 1:15. We have. I think we have buffer time.
No, actually, in the schedule it is the session 4, or we are making a break.
Yeah, it starts at 1:15, I believe.
The schedule it is 1.
No, so it's 1.
Okay.
Yeah.
I think yesterday there was a buffer, but today it has been modified. Hi, Dr. Sugnakar, Amit here, Amit Nargotra. Yeah.
Yeah, hi, Dr. Amit. I've sent just
yesterday even I saw there was a buffer for 15 minutes, but I think today it has been rescheduled.
Yeah, that's nice. Yeah, just one sec, please. Yeah. I think Dr. Batra is here. Jyotsna Batra. Yeah, I think I just seen her. Yeah. Amit is here.
Yeah, I'm here.
Yeah, thank you, Dr. Batra.
Also, my understanding was that we will have 15 minutes buffer.
Yeah, exactly. Yeah, let us take a quick 10 minutes break. I think because schedule the talks accordingly. Yeah, exact 10 minutes. Yeah.
Okay. Yeah.
Yeah. Thank you. Thank you. Thank you so much. Prash, can you unmute yourself?
Yeah. Hi, Subbu. Hi, Jyotsna. Yeah.
Yeah.
Hi, Prash.
Yeah. Thank you, Jyotsna, for obliging to our request. We look forward for your talk. Thank you so much. Yeah.
Did you? Sorry, Prash. Did you ask me something?
No, not really. I was just, thanking Jyotsna.
Okay.
For
Sorry.
obliging to our request. Amid our busy schedules. Yeah. Thank you so much.
Okay, I think like if Jyotsna is around, we can start the session, I believe.
As you suggest, yeah, I'm available. I'm just trying to figure out how to share my screen here.
Okay.
Because I'm not very versed with Microsoft Teams.
On the right side corner, you have something called as share content. You have a leave button in the right corner. And
is it? Do you already see it?
Yeah, could you please open?
Maybe full screen, is it? What do you mean by open?
Yeah.
And it always goes on left slide. This is interesting. Wait a minute. Does it look good?
You already selected it. We can say open option there on the right side. Right click and then.
Oh, you still don't see it?
Yeah, I can see on the screen. Yeah, InBix-2021. That's what has been written.
So on my screen, because I do not have an app, I'm using the web version.
Okay. On
I don't see anything on my. Wait a minute. Let me turn on the video also. Oh, it says you can share your video once you are done screen sharing. So I'm already screen sharing here.
You can open the.
Yeah.
So is it okay now? Because I have opened it, like, you know, it's in full screen on my screen.
I think there should be some.
Do you see them going? Maybe I stop sharing once, yeah? Okay. And then we see again. Because to me, it shows that everything is working. So I have a thing called Open Share Play, yeah? Okay. And then I do this screen share.
Click on your image, that what you see. Then you can, on desktop, you can keep this presentation and then you can open.
You mean entire screen? No, it still doesn't work. Screen. How does it look now?
Yeah, you can open your presentation now. Yeah,
it's already open. Yeah, do you see the title? Yeah.
Yes.
It's good.
Yeah, it's
excellent.
Yeah.
So my understanding is I will not see any videos, and you will also not see me, yeah, during the presentation.
Yeah. Yeah, full screen.
Probably there is a difference between. Sure. Okay, so can I start? It's full screen from my side. I hope you see it full screen.
No, yeah, now it's fine. It's perfectly fine. Can you just hide that? On that, you have stopped sharing and then you hide. Please hide that.
Okay, yeah, good. Okay. This is good now?
Yeah, it is good now. If you can
just... And do you want me to start, or are we taking a break?
Just 1 minute. Just give me a couple of seconds. I'll just try to give some introduction about Jyotsna Batra.
Okay, sure.
The audience will be looking forward to know about you. So it is our privilege that we are able to introduce you. Yeah. So thank you so much, Dr. Batra. To brief Dr. Batra, she is an associate professor at Queensland University of Technology, Australia. She has more than 15 years of research experience analyzing genetic variations for their associations with complex human health disorders and dissecting their functional roles with a view to identify new diagnostic, prognostic, and therapeutic modalities. During her PhD lecture in 2010 in the field of human genetics, she has published extensively demonstrating a positive association of alleles and several genetic variations with complex genetic disorders. In addition, she contributed large-scale genetic studies, including the Indian Genome Variation Consortium, which led to several papers, including Science and PNAS. That shows how voraciously she publishes the papers and in the best interest of the public. And she was awarded 2 highly prestigious awards and competitive NHMRC fellowships, a Peter Doherty Fellowship, and NHMRC Overseas Biomedical Fellowship, which further accelerated her career. studying genetic variations in the family of callicorin protease genes, the KLK3 protein, which is more commonly known to be, which is known as the specific prostate cancer antigen, is a serine protease cancer, which is an important clinical biomarker. And functional role in prostate cancer has been the major focus of our group. And apart from that, she's part of the entrepreneurship chair with regard to BioClose organization, where we have an CAPC called as Consortium, Cancer Prostate Consortium of India, which was incepted in late 2020. The main focus of CAPC is like the consortium aims to bridge the gap between geneticists and the clinicians working on prostate cancer. And that's where, like, as a part of the overseas member, Josna Batra was gracious enough to do her best for India. So with these few words, I welcome Dr. Josna Batra for her presence and sparing time with us and enlightening the students, the participants. Yeah, the podium is yours, Dr. Batra, please.
Many thanks for this opportunity today. I'm really pleased to be here, and this is such an interesting conference with an interesting program. So it's a pleasure to be able to talk today. As it's already been mentioned, that the focus of my projects have been on kallikrein variants. So today I'm going to talk a little bit about those with respect to prostate cancer. So just to give you a little bit of information about how prostate cancer's demography and epidemiology is in Australia, because this is where I come from, and it's really a major disease and #1 cause of cancer in men in Australia. Not just the #1 diagnosed cancer, but also it's the second most common cause of cancer death as well in Australian men. So our population in Australia is really small, but when you look at that small population, we still have around 20,000 men which are diagnosed with prostate cancer each year, and of which 3,000 men die. So it's so frequent that 1 in 7 men is diagnosed with this disease by the age of 75, and actually it's 1 is by the age of 85. So it's highly, highly prevalent cancer overall. And it's really important that when we look at this cancer in details, and we also, as previously mentioned, that I'm part of CAPC as well, and the goal of CAPC is also to look at the genetic component of prostate cancer. So is also my research. Interestingly, prostate cancer has a huge genetic component, which is approximately 50%. And it's 50% driven by the environment. So now when we talk about the genetics and dissecting the genetics of a complex disease like prostate cancer, it's important that we take several different approaches to do this. And one of the approach which we have been taking over the years is using the genome-wide association studies. So those who do not know about the genome-wide association studies, it's a very simple, straightforward analysis where we compare the genetic component of patients versus control. And it's different from sequencing because the technology used here is a chip-based technology where genetic variations, most commonly single nucleotide polymorphisms, are presented or genotyped using a SNP chip-based approach, and their frequencies are compared between prostate cancer patients and control in this case, because we're talking about prostate cancer genome-wide association studies. So using this approach over the years, we have been able to shortlist around 200 risk loci, which are associated with risk of prostate cancer in European or Caucasian population, because we haven't had much success so far because of the lower sample set in other populations such as Asian population and African American population, towards which the consortium of which I'm part, and I will describe a bit more about this a little bit later, has been now inclined to also look into other populations. So is CAPC is trying to also achieve the same. So using this approach, each risk variant or each single nucleotide polymorphism, which is associated with the risk of the disease, has very small risk estimate, because we are talking about germline variants here. But when we combine them together, the polygenic risk score have been shown to have clinical relevance and something which could be used for screening of the disease. Although the polygenic risk scores have been very interesting outcome for prostate cancer population screening, however, there have been some issues before we could clinically translate the genome-wide association studies data. And same is with the recent approaches which are about sequencing, because we can find variants which are associated with disease, but it's difficult to find something which is really a causal or a functional variant, because when we design genome-wide association studies, most of these the SNPs which we are using are actually linked SNPs, because they are present on the genome in the region which are easy to genotype. And we also take into account the concept of linkage disequilibrium. So one of the single nucleotide polymorphism here would be tagging several other SNPs in the gene, and most of these variants are present in the gene desert region or far away from a particular gene or the coding region of the gene. So once you have identified a SNP which is associated with the disease risk, there is a mammoth task ahead of us to identify what could be its functional role, what are the nearby genes it is regulating, whether it's a gene, protein coding gene, or it's a non-coding part of the genome like long non-coding RNA, or other unknown causal genes present nearby. So what is it actually regulating? So this then opens up a new concept of post-genome-wide association studies, or a post-GWAS as we call it. And this is a huge work because we have around 200 risk loci identified, and most of these are in the region of non-coding part of the genome. So we thought of overcoming this problem of GWAS by focusing on exon SNP chips. And when we analyzed the exon SNP chip, we actually were not able to identify anything which were significantly associated with the prostate cancer risk. So here again, I would emphasize that we are talking about germline variants. So we took an approach where we thought we cannot find anything in the exon using the exon SNP chip. And if we are only focused on the regions which are present away from the coding region of the genome, it is a huge task. Why not we focus on something which is easy to identify and could also give us the outcome in terms of the functional genome, functional variants? And this is where we started to look at miR-SNPs. Now, what miR-SNPs are, before I actually go into miR-SNP, I wanted to just present this cartoon, which is a very simplistic figure of what microRNAs are essentially. So microRNAs are translated also from the genome, as are the other typical genes. But interestingly, on the basis of sequence complementarity, microRNA binds to the complementary mRNA sequence. And because of this double-stranded structure formation, now the ribosomal machinery is not able to bind to this double-stranded moiety, and thus the translation can be inhibited. Sometimes this complex then can be attacked by nucleosomes and can be degraded. So one microRNA can regulate up to 100 to 200 genes simultaneously, because the sequence complementarity which is required is mostly around 7 to 8 mers, which is called the seed site of a microRNA. So this is why sometimes microRNAs are also called the master regulator of the genome, even though they regulate the genome at a very micro level. MicroRNAs are also secreted in plasma, and they are highly stable there. And this concept have been utilized in several studies for identifying the potential diagnostic biomarker or even prognostic tools. We also undertook a study where we were able to identify a panel of 4 microRNAs which can predict prostate cancer early. I'm not going to go into the details of this study because this is published in scientific reports, and you can go through it as well. Here I show you another signature which is also associated with prognosis of the disease, and 4 microRNA, and when combined with the PSA, can also predict the outcome of the prostate cancer survival in patient samples. But what we are going to talk about today is miR-SNPs. So as I previously mentioned, that microRNA and mRNA interact with each other in a sequence complementarity manner. And if there is a SNP present, either in the microRNA or in the target gene, especially in the seed region or in the complementarity region, which is the seed region of a microRNA. This SNP can then interfere with the function of microRNA. It can either stop or inhibit the binding of a microRNA to its complementary mRNA sequence, or even can create a new microRNA binding site to a target gene. So we started looking at these SNPs for their association with prostate cancer risk. In this case, we were specifically focused on the 3' UTR region of the mRNAs because we wanted to really identify the target mRNAs as well, and if possible, some of the important microRNAs which would be involved in prostate cancer. So again, this is a busy slide, and I will not get into the details, but this just gives you an indication of the sample set which we started doing our analysis in a cohort from Australia. So we did several recruitment studies over the years. Most important of these is Australian Prostate Cancer Bioresource, which has got 4 nodes all across Australia. And through this biobank, we have been able to collect samples for around 3,500 prostate cancer patients. And then the second component of this study was, of course, the healthy individuals, which we also recruited either through the Red Cross Blood Bank or via another project which we called as Queensland Men's Health Project. And this project was specifically to match our patient sample with respect to their age and postcode. So we approached the Electoral Roll Commission of Australia and got the information about men who are registered for Electoral Roll Commission, and were able to identify their age and their postcodes, and were able to then match it to our prostate cancer patient and recruit these healthy controls in our studies. But when we started doing this analysis in a smaller sample set of 2,000 patients and 1,000 controls or so, we realized that this sample set was not enough for us, because the risk estimates which we can identify through genome-wide association studies are pretty small, and we are looking for small effects, so we need a big sample set. Also, we need to take into account the patient heterogeneity, and thus a big sample set was required. So we then started analyzing these variation in a larger cohort. The data I'm going to present today is analyzed on 22,000 prostate cancer patients and almost similar number of healthy individuals, where we analyzed these MIR-SNPs through a part of the consortium known as the Practical Consortium, which is led by ICR London. And in this picture, you see Professor Ross Eels here, who actually leads this consortium out of London, and now is currently funding through NIH funding, and thus US is a bigger part of the consortium now and is led through there. So we were designing this new SNP chip called as ICOG SNP chip, and this is where we were able to present the 2,000 or so MIR-SNP which were identified by our group in Australia using the computational approaches. So we used 3 different software tools to identify potential MIR-SNPs, because these are all by prediction at this stage. And anything which was predicted by 2 out of the 3 tools were classified as MIR-SNPs and were then genotyped on the ICOGS array. ICOGS array are now commercially available, so anybody can buy the SNP chip and can use this this MIR-SNP approach as well. So what you are looking here is a Manhattan plot. So on the x-axis are chromosome number. Each dot here represent a MIR-SNP, and on the y-axis is the log p-value of association of these MIR-SNP with the prostate cancer risk. So when you look at the outcome here, the most significant association which we identified was in the 3' UTR of the kallikrein-3 gene. We also identified several previously, so there were a total of 16 significant MIR-SNPs which were associated with prostate cancer risk. So 22 MIR-SNPs in 16 genes. Several of these fall in the region which were previously identified by GWAS, while we were also identified were able to identify several new genes with this approach. The SNP which we are looking at now in VAMP8 was also associated with the risk of prostate cancer. So I will go through this data a little bit faster because this is also published data, but then towards the end we'll present a story which is not yet published, and we'll like to spend a little bit more time on that. So because there were still 16 genes in total, and we wanted to start analyzing them for their functional role in prostate cancer and also to identify relevant microRNA, we started to look at their expression profiling in normal versus tumor tissue. So when I say normal here, this is the adjacent tumor tissue, and the data you're looking at is a Grasso data set, which was at that stage was the biggest data set which was published. So of the 16 genes, 7 showed differential expression, and the 7 genes were highly expressed or overexpressed in the tumorous tissue from prostate cancer patient. The data I'm going to present to you today is on kallikrein-3 gene, because this is such an important gene for prostate cancer. It encodes for prostate-specific antigen, which is a current biomarker for prostate cancer. We have been working on all other genes as well, slowly. We have already dissected the functional role of MCAT, PDK1, MDM4, and VAMP8, and PDLM5 and TLL12. Other groups have worked and have also shown really important role of these genes for prostate cancer. So our approach has been taken forward by other groups as well and have been validated and has shown to have impact on the disease. So now going back to kallikrein-3, as I previously mentioned that the MIR-SNPs are based upon the prediction, and here what you see is the complementarity of the mRNA and the microRNA region, and these 7 to 8 nucleotides are the seed region of these particular microRNAs. So in this case, we predicted 3 microRNAs, 2 of which MIR-219 and 3162 seems to be binding when we have the T allele in the 3' UTR region of the kallikrein-3 mRNA, while 4278 was predicted to bind to the C allele. So the first task for us to validate this prediction analytical outcome for our MIR-SNPs, for the associated MIR-SNPs, the ones which were associated with the risk of prostate cancer. So the first experimental work which we undertook was reporter gene assay, and what you are looking here is that we cloned the 3' UTR of the kallikrein-3 gene. We also did a site-directed mutagenesis to have either T or C allele insert and transfected it with the microRNA mimics into prostate cancer cell lines. And very interestingly, of the 3 predicted microRNAs, only 1 was able to show effect in the reporter gene assay in the allele-specific manner, while the 2 other predicted one did not show us any significant effect. Then we also validated this outcome using the internal expression level or endogenous expression level of kallikrein-3 at the mRNA level and at the protein level. And as you can see, that overexpression of the MIR-3162 in prostate cancer cell line decreased the mRNA of kallikrein-3 as well as the protein expression level of kallikrein-3 in a time-dependent manner, essentially. So not just kallikrein-3, but there are several other members of kallikreins. For example, kallikrein-2 and kallikrein-4 expression was also downregulated when we overexpressed MIR-3162 in these cell lines. And not just this, androgen receptor, which is also a very essential component of prostate tumor growth, was also downregulated when we overexpressed MIR-3162. And we have to test androgen receptor because kallikrein-2, 3, 4 are all regulated by androgen receptor, and we have to put this concept forward that whether this is a direct interaction of MIR-3162 or whether it was down-regulating androgen receptor, and this is how it was also down-regulating other androgen responsive genes in prostate cancer. And to our surprise, it was also showing the down-regulation of androgen receptor. But then to validate our hypothesis, we also did the pull-down assays, where we transfected the biotin labeled microRNA mimics into the prostate cancer cell line, pull down the binding partners and look at their expression. What you're looking here at the mRNA level, but we are also sequencing to identify additional components or additional binding partners of this microRNA. But what was evident through this pull-down approach that MIR-3162 pulls down kallikrein 2, kallikrein 3, kallikrein 4, and androgen receptor. However, the other genes, which in this case can be considered as negative control for pull down assay, were not pulled down, which are the other members of kallikrein 3. Kallikrein family, and I will show you this family again to give you a clear picture. So this gave us an indication that this microRNA is an essential component of regulating prostate cancer related genes, including androgen receptors. So we also wanted to look at the effect of this particular microRNA MIR3162 on prostate tumor growth. So first thing we tested was expression of this microRNA in different cell lines, and as you can see here that its expression was highest in PC3 cell line, which essentially do not express androgen receptor or any of the kallikreins, giving us a very important indication that this microRNA could be responsible for differential regulation of kallikrein family and androgen receptor in different cell lines as well. Nevertheless, overexpression of this microRNA reduced the prostate cancer cell growth. This was an interesting observation that in the initial hours, it actually leads to slightly increased prostate cancer cell growth, but later on it decreased cell growth. What you're looking here is a three-dimensional colony formation. As you can see, this microRNA is highly effective in colony formation outcome for prostate cancer cell growth. So this seems to be an interesting microRNA. We were done the pull down assay for only subset of its substrates or its complementary mRNA, not everything. So in this case, we undertook a MS-based approach, and we looked at the change in the proteomic profile of the prostate cancer cell lines after overexpressing MIR3162, and interestingly, very important pathways such as oxidative phosphorylation, fatty acid metabolism, MYC targets, G2M checkpoint, and mTOR signaling were found to be regulated by this microRNA. So this present a very interesting outcome, and we took the similar approaches for all other genes which we have identified and I mentioned to you previously, which led us to the identification of few other significant microRNAs using this approach as well, and one of which we have already taken into in vivo models. And it seems to be highly effective when we overexpress that particular microRNA, which we are calling as prosmir, because I cannot disclose the ID of that microRNA as we are keen on filing a patent for it towards the therapy of prostate cancer. So overexpression of that microRNA abrogates the tumor growth in in vivo models as well. So this is where we have taken our microRNA work, but now going back to the locus of kallikrein, which I mentioned to you previously, and this is also a very interesting story, and I will quickly go through it. That after doing the GWAS, then we also took the fine mapping approach, which is essentially sequencing all the locus which have been identified by the GWAS approach or our MIR-SNP approach. So we started sequencing those regions as well. And as you can see here in this graph, that kallikrein 3 belongs to a family of total of 15 genes, kallikrein 1 to kallikrein 15 in total. 15 was discovered last in the opposite direction. This is why the position is different here. All present on chromosome 19, Q13. As you can see here, that in the 3' UTR region is present our MIR-SNP, but when we did the deep sequencing of this region, we were also able to identify exonic variants which were associated with prostate cancer risk, and also at a very highly significant level. One of this is present at exon 3 and another one is present at exon 4. The SNP which is present in exon 4 is a non-synonymous SNP, which leads to, so this is the position at the mRNA level, 536. It's a change from T nucleotide to C nucleotide, but because it's a non-synonymous SNP, it also changes amino acid sequence or PSA. So the sequence which is changed is the original at the 161 position is isoleucine, and when the SNP is present in those patients, the isoleucine is replaced by threonine amino acid. And as you can see here, odds ratios indicate that the frequency of this SNP is higher in healthy individuals in comparison to prostate cancer patients. It's highly significantly associated with prostate cancer risk with p-values of 10 raised to power minus 81. So this was an additional interesting outcome of the fine mapping study. And this analysis was performed in around 80,000 prostate cancer patients and 60,000 men. And with additional analysis, we have confirmed this in more than 100,000 prostate cancer patients using the approach and the practical consortium collaborative approach. But now in the next 5 or 10 minutes, I would just quickly describe what we have done to identify the function of this SNP, which is present in exon 4. So when we discovered the association of this SNP with prostate cancer risk, there was a conundrum that this is not a true association with prostate cancer risk, but instead the association is with the PSA. And as I previously mentioned, that PSA is a biomarker for prostate cancer. Its level is detected in the plasma, and this is how patients are initially screened. And the screening is very common in the US and the UK, and this is where most of the patient cohort in the consortium has come from. So the concept put forward was that this SNP has no functional role as such, because PSA does not have a functional role in oncogenesis of prostate cancer, but we see this association mostly because of the screening of the patients based upon PSA. So it's a mere association of the risk or a SNP with PSA levels, not with prostate cancer oncogenesis. So we tried to solve this problem by doing some functional analysis. So for this experiments, what we did is express the PSA into the cell line, PC3 in this case, which do not have any endogenous PSA expression. So we can look at the effect of overexpression of PSA from exogenous expression, so that we can then also compare this overexpression with other type of PSA. So whenever I'm talking about wild type PSA, which means it's the which is represented here as WT, is the common PSA sequence. T161 PSA represented in my next graphs are the single nucleotide polymorphism in exon 4, and it's the threonine variant. This is why it's represented at T161 PSA. A195 PSA is a positive control in these studies. This is an important position in the kallikrein 3 or the PSA protein, which is part of the catalytical triad. And in the previous studies, it has been shown that if you mutate this, then PSA is inactive. So this essentially is a positive control. And as you all know, that vector-only overexpression in the cell line is another control, which is used to compare, because you cannot compare your overexpression cell lines to the naive cells, because there will definitely be some differences just because of using the transfection methods. And in this case, the transfection methods, we use several different transfection methods for different experiments, but these are the stable clones, what you're looking here. So as you can see in these graphs, that overexpression of the wild type PSA increase the cancer cell proliferation as well as their migration, which is represented here by closing of the wound. However, overexpression of the threonine 161 PSA had no effect on the prostate cancer cell proliferation and their migration. We then also looked at this data in 3-dimensional analysis, and what you are looking here is an spheroid model, but interestingly what we see here is kind of an opposite result, because overexpression of the threonine variant seems to make PC3 cells highly aggressive in the spheroid model, which is also can be clarified using the compactness. So these spheres are no more compact, and they have high area. So this was a very interesting 3-dimensional analysis, which gave us outcome in the opposite direction to what we saw in the 2-dimensional analysis. And when we submitted this paper, we were asked to do a replication of this analysis in another cohort or another model system. And this is where then we use the prostate cancer organoid model system, where we overexpressed again the same PSA construct in these organoids, which are driven from Memorial Sloan Kettering Institute, and we have them in our lab now. And also in these models, what we could find that overexpression of the threonine variant actually had much higher number of spheroids, and these spheroids are widespread as well, as you can see from the area, and they have reduced compactness, showing again that these spheroids are quite invasive and are going very much in a direction of metastasis of the disease, if we want to compare this to the physiology of a human being. So this was a very interesting result because we saw the different outcome in 2D and 3D model. So to confirm this effect, we thought that we need to analyze this in vivo model system, or in fact, I would say that this was also a demand from the reviewer of the paper which is not yet published because this was sent to Cell, and they asked us to do so many experiments that for last 2 years we have been performing these experiments again. So when you see here the overexpression model, when we express the PSA wild type, the tumor started to grow really faster in a subcutaneous model of the in vivo, which is a model of the local tumor growth. While as you can see here, the tumor are pretty small. Also in these graph, you can see the same effect where the expression of threonine variant reduce the solid tumor growth in the local area. However, to our surprise, very interestingly, when we use the intracardiac injections of these same cell lines, and which is a model of tumor metastasis, we were very surprised to see the opposite effect. So the injection of the PC3 cell lines, which overexpress the threonine variant of PSA, highly metastasized to different, and you can see some of the graphs here. So this was a very interesting analysis again to get the opposite results for the local tumor growth versus metastasis. So this represents that the PSA SNP has got contextual role. While it is protective in the local tumor growth, it leads to high metastasis in the later growth area. And to explain these results, we used additional experiment analysis. For example, what you are looking here is that is at the human vein endothelial cells, which show you that when we have wild type PSA, the HUVEC growth to form the vessels, the blood vessels, is highly reduced, while we do not see any effect when we have the threonine variant PSA. We also generated the human bone model. Again, I will not have time to go into the details of this, but the wild type PSA as well as the mutant version of the PSA with threonine variant. What we look here is that on the humanized bone model, the threonine variants are highly invasive, and this leads to the concept that how this would be highly important with metastasis. Now, the last model I'm presenting to you here comes from the human data, and I previously mentioned to you that the wild type PSA, the C allele was protective against prostate cancer, but we then were also able to look at the out these patients for their survival type or genome had poor outcome with respect to prostate cancer survival. So this again proves our hypothesis that PSA has a, or the PSA SNP has a contextual role, and how is this contextual role can be proven is through if we can understand what PSA does. So PSA is a proteolytic enzyme which has got its normal function in semen liquefaction, but in prostate cancer it can activate, because it's a protease enzyme, activate or deactivate several other enzymes. For example, using IGF activation. It can lead to binding to IGF-PP3. It can activate pro-PSA. And what we could show using the synthetic PSA, that the threonine variant has highly reduced proteolytic activity against these peptide substrates, as well as for the several of the protein substrate. We also looked at the molecular dynamics study.
Yeah, Dr. Batra, sorry to intervene.
We see that the presence of the threonine.
Yeah, 2 minutes.
Yeah, it's this, yeah, almost. Yeah, it will not even take 2 minutes. So this can reduce the flexibility of the kallikrein loop. And this is how it affects the structure and stability of the PSA. Last but not the least, we could clearly show that the threonine variant is associated with the and then we're looking at the immunohistochemistry level, and it also associates with the PSA level in the plasma. So this brings us to the concept that the patients who have got this allele or the threonine variant of the PSA have highly lower level of PSA, as you can see in this graph, and these are those patients who would end up having highly aggressive disease. So we are already aware of these sort of analysis where patients who have got low PSA level end up with the poor survival outcome. So our hypothesis is that these patients who harbor these genetic variants would have worst outcome of the disease because of the reduced protease activity, the data which I have shown you today, but also because of the reduced protein stability and formation of inhibitor complex, and both these data I have not shown you today. And these patients which have got this threonine variant leads to an aggressive metastasis of the disease. And not just that, because of the we also did the clinical testing of this PSA by producing them in a sample set. We also could show that this PSA cannot be detected very well with the clinical existing clinical assays. So these patients also have the reduced detection of PSA. So these patients need to be specifically monitored who have harbored these genetic variants, because these are the ones which will be detected as low PSA, but at the end will end up having highly aggressive disease. So we plan to develop a novel antibody-based test, or we could even use the genetic-based test for the measurement of these patients in the clinic. And with this, I would end my talk here, which actually shows you a benefit of the GWAS association study, starting from the genetic association analysis, that how it could benefit patients in the long term if we undertake the fine mapping and functional studies to identify the contextual role of the disease-associated variants. So this is my group. In fact, I have several new people and funding agencies who have been highly supportive of this research. And with this, I will finish my talk here and will be happy to take questions. Thank you.
Yeah, thank you so much, Josna Batra. There was a very scintillating talk, and the way you focused on starting with the GWAS, then talking about the variants that related to the PCA, then trying to talk about the functional validation. We can take a quick 1 or 2 questions. I have my personal question as well. I'll just ask that. One of the participants is asking, there are several androgen receptor variants like ARV1, 3, 5, 7, 9 are mainly associated with aggressiveness of PCA. So which would be more preferable to target with recent microRNA-based mRNA targeting approach? Or these could be a secondary effects associated with undergoing with mRNA therapy?
Yeah, I absolutely agree. So in our study, we have not looked at the AR variant, but I'm aware of other groups who are specifically investigating. We need to look at whether we have these variants have got similar 3' UTRs, some of which, example, V7 ends up early because of the presence of a stop signal. But if they still have the same UTR, the same microRNA could target all of these variants. So this will be an interesting analysis to do a comprehensive and detailed analysis, essentially. But as I have previously mentioned, that microRNA-based therapy could be a bit complicated because it targets several, it has several target genes, and some of these could be oncogenes, and some of these could be also protective against the disease. So we have to make a very good balance when we are taking this approach, and a detailed analysis in several models is required.
Okay.
Before we can use any of the micro.
Sure. Thank you so much. I have a question, like whether the GWAS data that is publicly available in GWAS Central or Practical Consortium, is it the GWAS data?
Yes, it is. It is publicly available. So data on, I think, 172,000 patients in control, which is genotyped on OncoArray, is publicly available. Yeah, I myself have actually also downloaded it through the NIH databases. So yeah.
That's wonderful. Like, thank you so much. Thank you for your time, and looking forward to interact through our CAPSI project. Yeah, thank you, Dr. Batra. Thank you once again.
Excellent. Thank you.
Yeah, I want just a group photo, a quick group photo, please. Could you please switch on your cameras, all of you? Switch on your cameras, please. So the opportunity we get it, events like this. Yeah, please, everyone. Yeah, there's something called as a together mode, where there is a lot of scenic beauty that is being seen in MS Teams. So I request everyone to come over here. Yeah, please. Others as well. Yeah, please. There is nothing to feel shy. Yeah, please. It's a rare opportunity. It's really good to be part of. Taking a good snap, right? Yeah. Smile, please. Yeah. Thank you. Yeah, thank you so much. It's really a pleasure listening to your talk once again, Batra. Yeah, thank you so much. Now I give.
Thank you.
Yeah, thank you so much. Now I ask the co-chair, S. Nagamani, to introduce our next speaker, next invited speaker, Dr. Amit Nargotra. Yeah, it's all yours, sir. Please.
Thank you, Dr. Sugu. So the second invited talk will be delivered by Dr. Amit Nargotra, and he is a bioinformatician with over 15 years of research experience, especially in the area of computer-aided drug design and discovery. And he completed his PhD from BITS Pilani, and later he did his PhD in, he did his engineering from BITS Pilani, and later he did his PhD in bioinformatics. Currently, he is heading the Discovery Informatics group in IIM Jammu, and the major research activities in his group includes the identification of novel scaffold for drug discovery, especially for the cancer. And he has established a state-of-the-art molecular modeling facility at IIM Jammu, and he is also managing the institutional compound library. And currently his group is mainly working in the developing various computer-aided drug design models and their applications, and eventually that his group is collaborating with the experimental to test those molecules. And his group is mainly working very keen and closely collaborate with the medicinal chemist, natural product chemist, and pharmacology group. And he has more than 40 sci-index publications and 6 patents in his credit. So with this brief introduction, now I request Dr. Amit to deliver his lecture. Sir, over to you.
Thank you, Dr. Nagmani.
Thank you.
It's a pleasure to be here, and thanks for inviting me to this wonderful conference and sharing my research experience through this platform. I'll just close the video and share my screen. Please let me know if the slides are visible. Yeah.
Sure, sir.
Yeah, is the slide visible?
Yeah, please go ahead, sir.
Yeah. So as the title of my talk, it says the role of cheminformatics in drug discovery and obviously the way forward which we are looking into. I would be sharing the experience over here being like as Dr. Nagmani mentioned that I am an engineering by training, and it had been an over around 20 years journey into this bioinformatics and cheminformatics drug discovery area. So I would be just highlighting, giving you the glimpse about the activities that we are carrying out in our institute IIIM, which is a purely drug discovery institute. The mandate has been, and we are lucky to have several divisions which actually right from the beginning. In fact, I would these are the broad overview about the various divisions which we have here, and all of them, they are working in very close collaboration. And I feel as a bioinformatician or as a cheminformatician, I'm lucky to be here in this institute in a close collaboration, working with the chemical sciences, chemical technology, biotechnology, pharmacology, like right from the beginning of the drug discovery process till the end, like QCQA and CMC studies of all the things. And now we have a very good established GMP, CGMP facility where we make the capsules and tablets, though it is not Schedule M approved, it is right now WHO approved, and we do the studies and make tablets for the proposed phytopharmaceuticals which are actually proposed from IIIM. So as I mentioned that we right from the beginning in the natural resources regarding botany and microbiology, we have a very well established division over here, which actually collects all the natural resources which are required for the drug discovery process. And then a very well-established chemical sciences group where we have the natural product and synthetic chemistry, medicinal chemistry division, and we are actually working in very close collaboration with them in the general pharmacology also, where we do the validation part. So the point is that I want to focus here is that whatever we are working on, it is not that we are working in a standalone cell. We are being very well collaborated with these divisions, where whatever we are predicting, it is getting validated right immediately. So that sometimes it is actually I would say not very encouraging also, because immediately sometimes we predict a lot of things, and whether it goes to the wet lab or not, that is actually to be seen. So my talk actually I would be dividing into probably three components. The first part is that the compound library which we are actually maintaining, as Dr. Nagmani also mentioned in the introduction. So that has been a very important resource for carrying out all our discovery informatics programs here in this institute. And I would highlight that how we are utilizing that compound library. Then a brief highlight about like how we have been using these compound library over various disease areas, which relates to cancer, inflammation, and other parts. And the last part, just for a deliberation part, because what exactly we have been doing and the world have been doing, whether that has brought any really significant change in the drug designing, or we are just moving forward, and what exactly we need to ponder upon to really have that effective drug discovery program using this cheminformatics as a tool. So primarily, I'm as I mentioned you that like in the drug discovery program, if we just give a quantitative slide, like where we can actually say it's a very highly optimistic slide, where we say that okay, in 10 to 15 years we will have one compound approved. If we start with around 10,000 compounds, or now we are actually in the data age where we have been talking about billions of compounds and all. So it is definitely not a very it's definitely a very optimistic proportion where we talk about that one compound will be approved after 15 years. But this is just to actually speculate that what exactly we are talking about, that okay, even if we start with 10,000 compounds, and probably sometimes even at the end of the lifetime you are not able to discover a drug. But this is just a quantification of the drug discovery program. And to highlight that basically we are focusing as a bioinformatician or as a cheminformatician, we are focusing here on the initial 8 to 10 years, which is actually the discovery program. And now definitely we want this particular time period to reduce drastically with several tools and so many techniques available. And obviously we'll be using the molecular modeling approaches, whether these are structure based or ligand based, for achieving this objective. And as I mentioned, as a molecular modeler, I'm sitting at a place where definitely we have a very good state-of-the-art infrastructure. That is what is required. We need obviously relevant software and infrastructure and other online resources. Apart from that, definitely the medicinal chemistry, where we are actually we have a very good medicinal chemistry division over here, which actually goes on for the synthesizing, and we are working in a close collaboration for all the discovery programs. We are working in a very close collaboration with these chemistry as well as the pharmacological evaluation. And apart from that, as I mentioned, that we have a very good resource of the compound library, which is close to 1 lakh compounds within the institute, which we are actually having inside the institute, and we are maintaining that. I just gave a brief about that compound library. As the Triple M, we are actually working in the disease area of infection, cancer, Alzheimer's, inflammation, pain management, and resources obviously in the form of compound repository. It is actually the starting point for any discovery program, so that we do not have to, obviously we screen several other zinc library and other billion compounds, but obviously to get those compounds, sometime it take months together to get those compounds within the institute. So this we are in, and apart from that, like the compound repository is not only on the commercial library. We have all the medicinal chemistry programs which are going on, and there are intermediates which have been produced, which are producing in that. We are even maintaining those compound libraries also, those compounds in the in our repository. And a very important resource is the natural product resource. So we have close to around 700 compounds in this natural product library, which is a unique, I would say, a unique resource. Because even when we tried to procure these compounds, some natural products from the external vendors, initially there was a lot of quotes that okay, we have around 1,000-2,000 compounds, but when it come down to purchasing that, they only could actually supply only 50 to 100 compounds. So we have close to 700 compounds which are actually within the institute, which is accessible anytime for all the discovery programs. And we the third category is the externally procured drug-like compounds, where we have applied drug-like filters on the existing commercial library so as to prune down the number to a reasonable one, and then we have procured these compounds, and we are continuously, after every 2 or 3 years, we are enriching this library of ours. So as I mentioned, we have a very important pure natural product repository with us, close to 700 compounds in this repository, which is growing gradually now. And like it has been now maintained for the last 10-11 years, we have the several different categories of steroids, phenolics, terpenes, coumarins, lignans, and several other classes. And some of them are actually totally new compounds which have not been reported earlier anywhere. And that is actually, the credit goes to our natural product chemistry division, who are actually regularly isolating these compounds from the natural resources, whether it is plant or microbes, or even marines also. Nowadays we have started. The other repositories, we call it NCE, though it is not the NCE in real sense of drug discovery, but these are the new chemical entities which are produced within the institute as a part of various several medchem projects which are going on within the institute. And all these compounds, usually what used to happen was that whenever a PhD student, these areas for